Okay, so it should have started right now. So this is my great pleasure to start this new ERC IDEM workshop, supposedly based in Bordeaux, but because of the COVID-19 situation, this is a virtual meeting, which is also very nice because I guess not all of you could have come to Bordeaux. So this is really nice to have you all here on the screen and gathered for this second meeting. Last time, as you know, we had Scott Gilbert, Paul Doolittle, and John Roughgarden, and we talked about Holobionts. This is on YouTube available. And today we have our second meeting with a mixture of philosophers and biologists. I'm sure we will learn a lot. Our first speaker is Paul Griffiths. Paul Griffiths is professor of philosophy at the University of Sydney. He's an ARC laureate fellow. He's the domain leader for society and environment at the Charles Perkins Center, where he heads the theory and method in bioscience group. So a very interdisciplinary group embedded in the Charles Perkins Center with interest in biology and medicine. Paul is undoubtedly one of the most innovative and central figures in philosophy of biology. He's the author of many papers, including on psychology, developmental system theory. Also, Paul is the author of several books, including the now classic Sex and Death with Kim Sterelny, and also classics Genetics and Philosophy, an introduction with Carola Stotz. Paul recently worked on causal specificity in biology and also on the naturalization of health and disease. He's also been very interested in issues around individuality and pregnancy. And today, Paul, if I'm right, you're going to present something on that individuality and pregnancy topic. So this fits our general question, which is, as you know, biological individuality, microbiota, and holobionts. So Paul, the floor is yours.
All right, I'm sharing my screen. Okay, can everybody see that?
Yes.
Yeah, all good? Okay. Yeah, so this talk was actually inspired by Elsalyn Kingma's work, who some of you might have come across more through the philosophy of medicine than the philosophy of biology. And a few years ago, Elsalyn did that classic thing to get a debate going in philosophy, which was to point out that everybody was just implicitly assuming a view and had never really thought about it. And she gave this view the container view of pregnancy, the idea that there are two individuals, a mother and a fetus, and the fetus is inside the mother, contained within her. And so what she then did was to propose that this view, which nobody had ever really thought about, but had basically assumed, isn't true. Instead, she put forward what she likes to call the lady parts view, the view that fetuses and mothers are overlapping individuals. Okay, so that a fetus is at some time and in some ways part of its mother. And these papers have been, I think, very influential, controversial, but they're defended on primarily metaphysical grounds. So the way that Elsalyn has made the argument is by looking at the metaphysics of parts, so-called mereology, and arguing that things that philosophers say about parts and wholes ought to lead them to this view of pregnancy. And so I was very interested in that, and I wanted to know, well, from a more scientific point of view, if we look at just biological accounts of individuality effectively, then what sort of support do we get for Elsalyn's view? So that's really what this talk is about. Now, the view of individuality that I'm drawing on is the one that came out of one of the last bits of work that I was able to do with Carola. So we published a couple of papers in 2018 on process theories of human nature and process theories of development. So what I'm going to do is to present a process philosophy theory of individuality and use that to sort of exemplify that view, I guess. So for the purposes of this audience, maybe make it more clear how that view works and what sort of consequences it has by applying it to the case of pregnancy and whether or not the fetus is a part of its mother. So we're starting off here from process theories of the organism. I'll talk a little bit about those. Then I'll talk about the problem of individuating developmental processes. So if you're going to treat an organism as a process, how are you going to distinguish one process from another? And then I'm going to apply that to fetuses. I'm going to introduce the idea of extended processual phenotypes. And if I've got enough time, I'll look at this interesting question, whether or not you're entirely yourself after pregnancy. So I didn't sort of, recently convert to process theories of the organism. As you can see, this photo was taken many years ago when Russell Gray and I were still young and I had hair. Way back in the 1990s, when we were just beginning our first academic jobs, we realized that the best way to think of a developmental view of organisms was to think of an organism as a process. So instead of thinking of an organism as a chunk of matter which comes into existence, stays in existence, and then goes out of existence, we think of an organism as a series of events, a series of things that happens. That period we like to call these life cycles quite a lot. So in this 1994 paper, we argue that evolution is best construed as the differential replication of total developmental processes or life cycles. So, I introduced the word life cycle because when we talked about developmental processes, there's a natural tendency for people to think that we were talking about only the early stages of the organism's life history. Of course, we meant the whole process between the organism's creation and its extinction. And again, slightly longer quote, we think of an evolving lineage as a series of cycles of developmental processes where tokens of the cycle are connected by the fact that one cycle is initiated as a causal consequence of one or more previous cycles. Okay, so the events that make up this developmental process are developmental interactions, events in which something causally impinges on the current state of the organism in such a way as to assist the production of evolved developmental outcomes. Now, I've also put in a couple of papers there, a little argument that Russell and I had with Kim Sterelny and some of his collaborators. They were trying to argue that a developmental view of organisms could be accommodated within a kind of selfish gene theory by allowing there to be non-genetic replicators. So you'd have a sort of selfish gene and epigene and blah, and this and selfish that and the other, big family of competing replicators, and that would be enough to accommodate developmental views of organisms. And Russell and I replied in a paper to that. One of the points we made was that the idea of a selfish bird nest trying to replicate itself is actually very clumsy, as opposed to our view in which the nest is not part of the bird, but the interaction between the bird and the nest is part of the bird's life cycle. So we argued that the process ontology is a more natural way to represent straightforward biological common sense once you recognize that organisms are developmental systems which are embedded in and draw on their environment. Okay, you get around a lot of problems by saying, don't worry about the limits of the physical object, worry about which events are part of the process. Now you'll notice that we talked about events in which something causally impinges on the current state of the organism, not about gene-environment interaction. And that's an old theme in developmental approaches to organisms. It's something which Danny Lerman famously insisted on in his seminal 1953 paper. The interaction, says Lerman, out of which the organism develops is not one, as is so often said, between heredity and environment. It is between organism and environment, and the organism is different at each stage of its development. So the point that Lerman was making here was that talk about gene-environment interaction is a statistical abstraction. Interaction in a developmental biology sense is between the organism at any state in time, which includes its genome in a historically modified state. So the genome in a particular cell with the particular modifications it's accumulated so far in the life of the organism is just one more physical part of the organism, which then interacts with other physical factors, namely with some things which get classified as the environment. So the kind of real causal interactions are not G times E. The real causal interactions are the organism, including many modified copies of its genome, interacting with particular physical factors in the environment. On the other hand, it's often a lot easier to strategically adopt a gene-centered perspective because so much good research is done from a gene-centered perspective that if you want to just piggyback on good empirical research, it's often easier to kind of idealize, let's just pretend that the only things that are going on is genetic heredity and gene expression. So what I'm going to do is to focus on a developmental process or a life cycle as a series of events consisting of changes in states of gene expression. Okay, so that's going to be sort of a simplified way of pulling out some bits of the organism's life cycle that we know a lot about so that I can talk in a more concrete way. Okay, so given a brief introduction to treating organisms as developmental processes. So here's some quotes from the paper that Corolla and I published a couple of years back on the problem of individuating developmental processes. We say developmental systems theory needs to give an account of the gen identity, the generative identity of these processes. It needs to say where one developmental process ends and the next begins. And then we point out that this problem arises in a dramatic form when organisms have alternating haploid and diploid phases of comparable length, cases that Peter Godfrey-Smith has been telling philosophers to pay more attention to for some years. So if we've got for example, a haploid and diploid phase with a substantial and active haploid phase, then we want to know whether or not this is two life cycles or one life cycle, and we need some principled reason for choosing to say one or the other. Going on, so the principle of gen identity of a life cycle also needs to explain how a life cycle could consist of different series of events from one generation to the next. Okay, so you can't identify life cycles using similarity because you want to have lineages. One just does in biology, have lineages consisting of highly variable life cycles. What happens in one life cycle isn't the same thing as what happens in the next life cycle, and yet they're both examples of the same life cycle. So we say that problem arises in a dramatic form when species have a range of substantially different ways to get from conception to death. So think about the idea of, this is facultative pedomorphy. So we've got a creature that can either go through or not go through metamorphosis. So it can become sexually reproductive either in its larval form or what we normally think of as its mature form. So we're going to have a lineage of life cycles consisting of these two radically different life cycles succeeding one another, giving birth to one another. And we want to be able to talk about them being of the same kind. We want to be able to talk about one life cycle out-competing another life cycle when that life cycle has this disjunctive form. It can come in this either-or form. And as Corolla and I say, although that's dramatic, the same issue arises in principle whenever an organism exhibits adaptive phenotypic plasticity, so that successive life cycles in a single lineage do not contain the same developmental events. So we want to be able to say, what are the limits of a life cycle? Where does it begin and where does it end? We want to be able to distinguish one life cycle from another. Another important example is if we've got a an organism with a parasite, we want to be able to distinguish the series of events that constitutes the life cycle of the host from the series of events that constitutes the life cycle of the parasite, even though those two are a tightly coupled causal system. Okay? And we want there to be able to be the same life cycle, but to manifest on different occasions as a different specific series of events, and yet to still have a shared identity. So that's the same life cycle going down different alternative pathways of that life cycle. Okay. So what kind of principle can do that? Well, what we suggest is that there's actually a conventional piece of biological theory that does the job, which is life history theory. So Corolla and I said, for more quotes, a series of developmental events is a single process because they serve a common evolutionary goal, namely to maximize the representation of cycles descended from them in future generations relative to variant cycles with which they compete. We can draw on conventional evolutionary theory to make this suggestion a little more precise. An individual life cycle is a token of a life history strategy, and that strategy is its telos and its principle of gen identity. And I just have an antechinus there because it's just a cool example of life history theory. One of these Big Bang mating strategies. So I'm not going to do for this audience my standard, 5-minute introduction to life history theory. I'm just going to assume you guys are all familiar with life history theory. So what do we want to do with life history theory? What we want to do is to say that life history theory embodies a powerful principle of gen identity, the identity of processes, because the evolutionary rationale for the choice of strategy at each life history stage is conditional on what choices have been made or will be made at the other stages. A life cycle conceived of as the implementation of a life history strategy is held together by the trade-offs between its stages. So it's the fact that life history strategy is designed to capture all of the trade-offs between how an organism is at one point in its life and how it's going to be or has been at other points in its life, that allows it to unify the timeline of a developmental process. So I think it gets a bit clearer what we're doing in the next quote, where we say, if these events were not part of a single life history serving a single Darwinian telos, and we discovered at Bordeaux a couple of years ago that sometimes it's easier to say finalité than telos to get across what I'm talking about here. If these events were not part of a single life history serving a single Darwinian telos, then they would not trade off against one another in this way. It makes sense for me to accept an elevated risk of cancer in later life in return for my increased reproductive success, but not for your reproductive success, unless that's discounted by our coefficient of relationship. So the idea is that events which are part of a single life cycle trade off against one another in terms of their downstream effects on fitness in a way that's different from events in two different life cycles. So what you do has implications for what's the fittest thing for me to do, but the form of the trade-off between those is different from the trade-off between me doing two different things at two different points in my life cycle. Okay, so the events that make up my life cycle are events which trade off against one another in this distinctive way. That's the thought. Okay, so suppose we then, to try and make that clearer, apply it to the case of which life cycle is the very early embryo a part of. And what I want to argue is that from this perspective, the origin of the individual, the new individual, is not coterminous with the origin of the new genome. Okay, so forming the new embryonic genome doesn't constitute the beginning of the new life cycle. The reason for that is that the very early stages of the embryo are controlled by the maternal genome. So in humans, the initiation of embryonic gene expression occurs between the 4 and 8 cell stages. And I've just got a nice quote here from a review article that at this point, the embryo is the phenotypic expression of a stored maternal program that will be used to direct the completion of meiosis, events of fertilization, initiate cleavage, and perhaps mediate the activation of the embryonic genome. So the idea is that these events that are occurring in the early embryo, because they're the expression of maternal genes, will trade off against other decisions made by the mother in a way that is typical of the trade-offs within a single life history. So that early stage fetus is simply an extended phenotype of the mother. And so what happens there is part of the mother's life cycle, not part of the embryo's life cycle. Now that's very early in the embryo's development. It's a lot earlier than the kind of stuff that Elsa Kingma had in mind when she tried to argue that embryos were parts of their mother. But if we go on a bit longer, so these are quotes from one of David Haig's papers on maternal offspring conflict. And here's the quote. So Haig writes, "The maternal fetal unit can be conceptualized as having 3 haploid genomes: a non-inherited maternal genome absent from the fetus, an inherited maternal genome shared by mother and fetus, and a paternal genome absent from the mother." And then I'm emphasizing my emphases, of course. He goes on, "The conventional genetical division of this triploid entity into diploid maternal and fetal genomes, each of which functions as a coherent whole, involves presuppositions valid for most but not all loci, that genes expressed in mothers are unable to discriminate among offspring on the basis of offspring genotype, and that genes expressed in fetuses have the same effect whether inherited from mothers or fathers." And so I've just put in the figure of the IGF2 gene because this is one that's known to have parental imprinting. So Haig's idea here is that for loci which can overcome these 2 presuppositions, which don't confront those 2 presuppositions, we've got the possibility of the genetic interests being better conceived as the genetic interests of 3 haploid genomes than the genetic interests of 2 diploid genomes. So the fetus now becomes a site at which multiple genomes act. Okay, so the fetus is not an expression of its own genome, but it's the phenotype of its own genome, and it's the extended phenotype of maternal and paternal genomes. Okay, so the fetus is a contested kind of battleground. So then we ask, well, in that case, what does that say about the evolutionary individuality of the fetus? I wanted to, maybe I'll, okay. So, so we can apply a genetic conflict perspective in this way and talk about whether or not what's happening in the fetus serves the evolutionary interests of any of these, which one of these 3 genomes has its evolutionary interests served by what's happening. Another way to think about it is some event in the fetus the event which is the optimal choice from the point of view of one or more of these 3 haploid genomes? So that's a genetic conflict perspective. What I then wanted to do is to pair that up with a perspective on causal control that came out of earlier work that Carola and I and various other collaborators did. So this is work on how to attribute causal responsibility for an event such as the turning on or turning off of a gene in an organism. So as well as asking whose evolutionary interests are served by a particular event, we can ask of that event to what extent it's under the control of some variable, such as a locus in a particular genome. So we can look at an event, we can ask, is that event of gene expression under the causal control of a locus in any of those 3 haploid genomes? Now, one way to measure causal control was developed by Arnaud Pocheville and used in earlier work from our group, prescribed in these papers. It's very simple. It's the, this is down here is our specificity measure. And what it basically does is to measure the mutual information, I, between a causal variable and an effect variable, where the causal variable is not allowed to vary, but instead forced to vary by experimental intervention. So the hat operator means the value of the variable is determined by experimental intervention. And where this is conditionalized on all of the other causally relevant factors, which we've clamped with an intervention. So basically it says the extent to which one event depends on another can be measured by the extent to which, holding everything else constant, knowing about that event allows you to know what the, sorry, no, that's exactly what I shouldn't have said, because that would be using a conventional mutual information measure. So starting again, the extent to which one event is under the control of another is the extent to which, given everything else being held constant, if you have control of the cause, you have control of the value of the effect. Okay, and we can measure that using mutual information under intervention. So take the figure A representation up here, standard way of representing gene regulatory networks. If at some point in time, gene X is switched on, and we want to ask to what extent gene X's state of expression is under the control of gene Y or under the control of gene Z, we can do that by generating a table of probabilistic dependencies between these variables, by manipulating them so that, well, the classic way to do it would be to take every possible combination of variables and generate a maximal entropy distribution for each variable. So we force every variable to all of its possible values for each value of the other variables. So you generate a table of probabilistic dependencies between the variables, then we measure this, the specificity measure between the 2 variables we're interested in, and we get a measure of the extent to which whether or not gene X is in its on state is under the control of gene Y or under the control of gene Z. Okay, so that's just a piece of technology. If you don't like that way of talking about, or you don't agree with that approach to thinking about when one variable is under the control of another variable, you're welcome to plug in whatever your preferred way of talking about causal responsibility of one variable for another is, and that should fit into the rest of the apparatus I've set up here just fine. But the reason I'm talking about this is because, as well as having a way of thinking about whose evolutionary interests are served by a particular event of gene expression, I want a way of talking about which organism or which genome had control over that event of gene expression. So we can causally attribute it to variables in another genome or another organism. Okay, so if we put these 2 ideas together, we can introduce what I'm calling extended processual phenotypes. So an extended processual phenotype is just applying the extended phenotype idea to events where organisms are conceived of as series of events, and a series of events form part of a particular organism because they represent that organism's life history strategy rather than some other organism's life history strategy. So if we consider a fetus in that strategically idealized way I talked about earlier, we consider a fetus as a series of events of genome expression. Okay, so we reduce a fetus to a time series of states of expression of genes. We can ask to what extent those events are under the control of variables in each of the 3 haploid genomes described by Haig. We can also ask whether the actual state of genome expression at some particular actual state is optimal in terms of the reproductive interests of a gene token in one of those genomes. And I suggest that we would expect these answers to be congruent. We'd expect that events serve the reproductive interests of a genome to the extent that that genome has causal control of that event. So the obvious place to apply this is just the sort of place that David Haig has applied his genetic conflict perspectives, namely to gene expression in the placenta. So we find a tissue which we're already thinking about as a battleground between 2 genomes. And then we might expect that to the extent that one genome has causal control over gene expression in that tissue, the state of gene expression in that tissue will be closer to the optimal value from the genetic perspective of the genome that's got control, and further away from the optimal value from the perspective of the genome that doesn't have control. Okay, so tentative applications to thinking about whether a fetus is part of its mother. As I've said, the very early but post-fusion fetus, 1 to 4 cell stage, seems to be just an extended processual phenotype of the maternal diploid genotype. It really is, that early stage fetus is a part of its mother because the value of the phenotypic values we see in it are entirely under the control of the maternal genome. Then we might think about parental imprinting of genes. Well, if you look at parentally imprinted genes and you apply that specificity measure to ask whether or not the state of expression of the gene is under the control of the 2 haploid parental genomes or of the offspring genome, I think you'd expect to find that it's substantially under the control of the parental genomes, okay, and therefore you'd expect to find that its actual value is one that is at least closer to the optimal value for the parent than you'd expect if it was entirely under the control of the fetus. Now, the point I want to make here, which for me is really interesting anyway, is that the fact that the DNA coding sequences which make a gene product are in the offspring genome is not very important. So if you want to say, is that event part of the life cycle of the offspring, it's not terribly important where the DNA coding sequence from which the product was made is located. The real issue is where causal control was exercised in order to have that gene expressed in that tissue at that time. Okay, so causal control is what matters when determining which evolutionary individual is an event part of. Okay, not some physical constitution thing like where did it come from in whose genome. Okay, I think the parental imprinting of genes makes that clear, right? If a gene is switched on in the embryo in a way that is the result of something of variables in the maternal genome, and the expression state that results serves maternal evolutionary interests more than offspring evolutionary interests, or represents some compromise between them, then what's going on there is part of the mother. Okay, or at least it's part of. And remember that the perspective that Kingma is pushing is not that the fetus isn't part of the fetus; it's that there are overlapping parts. Okay, so she's trying to get this idea that some parts of the offspring are also parts of the mother. They overlap. And I'm taking a kind of processual event center perspective. Some events that occur in the offspring are events which are part of both the offspring's life cycle and of the mother's life cycle. They're overlapping parts in this processual event ontology. Okay. I also a little point that I've been very gene-centered here because it allows me to draw on lots of actual biology that people have worked out. But numerous other causal pathways exist by which mothers may exercise causal control over what happens in the fetus. So we have another quote from the review article I used. It would not be unexpected if the genetic program of the embryo were found to be assisted in early development by maternal paracrine factors. Okay, so it's not just making the point that my decision to do everything in this gene-centered way is just a strategic decision. I think all of these ideas would generalize to the whole developmental system. Okay, so I suppose I just have time maybe to do, are you entirely yourself after pregnancy? I think it's kind of clear that you're not entirely yourself during pregnancy. So we look at the literature on the placenta, and which is very much dominated these days by genetic conflict perspectives. So the idea that the life history of mother and fetus contain shared events, which are sites of potential conflict, I think is fairly straightforward. I'll just also, though, add there is this very speculative paper in Bioessays a few years ago, speculating that fetal maternal microchimerism might provide pathways by which offspring maintain extended phenotypes in mothers. So it could be the case that even after pregnancy is over, you're still not entirely yourself, which is a perspective that from a purely phenomenological point of view has appealed to some of my female colleagues. Okay, so just acknowledging our funders, and those are all the lovely members of our research group, Theory and Methods in Bioscience. And that is it. Very good.
Thank you very much, Paul. I'm trying to upload. I don't know if it works.
Yes, we can see your hands. Very good.
Okay, so thank you very much, Paul. Very, very good talk. We have 18 minutes for discussion. So who would like to start? As you can see, there's a way of raising your hand in, I guess, no, how does it work? I forgot. Let me see. You should be able to, oh yeah, Javier.
Yeah, it's in the participants thing. So you go to the participants, you come. So thank you, Paul. I really like the talk. I wanted to ask you, because it's very interesting, the perspective you're putting forward about pregnancy and individuality. But I wanted to ask you whether you cannot distinguish different stages during that process, because it seems clear to me, and there are some articles out there that say that, for example, before and after implementations, there seems to be a very strong change. Even some people have characterized this as an Aristotelian substantial change of what happens there. Once totipotency in stem cells occurs, implementation occurs, and then you have the three layers that give rise to the organism, it seems to be slightly different. So can you, with your perspective, distinguish stages during pregnancy?
Absolutely. I think that's what makes it richer than the philosophy of pregnancy literature that depends on mereology and metaphysics. I think what Elselin has, that go away. What Elselin's done is to put on the table the possibility in a kind of metaphysical way of mothers and fetuses, mothers and offspring, in fact, sharing something, being overlapping entities. I think once you plug in what we actually, know, we know quite a lot, particularly about the placenta. So I think once you plug in those details, I suspect it will be disappointing for some of the existing people in philosophy of pregnancy, because it's going to turn out that the period in which the fetus is literally a part of its mother is very short. And after that, it's more that there are many events which are sites of contestation. They're both part of me trying to do stuff and part of you trying to do stuff. So it will be, I think, less, it's certainly, I suspect, not going to be as attractive as some of the views that have been defended on a purely mereological basis. But I think that's what makes it so much richer. And in fact, it means that, new discoveries about what these various genomes are doing will actually change our views on the identity of individuals, which I take to be a much more richer and interesting kind of research program. So yes, you're absolutely right.
There's a question from Tyler.
Hi there. There's a lot of noise in the background, so sorry if it comes through. Thanks for the talk. There was a part later in the talk where you said that you thought that the sort of causal sense of control and the sort of adaptive life history-based sense of control should line up.
Yeah.
So I have a worry about that. I don't know if it's an objection to the view or just biological variation. But it seems like because a control in the adaptive sense is sort of an evolutionary outcome, it's something that needs to evolve over time, there's going to be lots of variation in how much maternal control there is in the early embryo. And it seems like the stuff you were talking about later, like conflicts between genomes, sort of meiotic drive things seem to come up in my mind as cases where the embryo, maybe the paternal genome, could evolve forms of subverting control. Yeah. But it still seems like in meiotic cases, the sort of causal sense of control comes away from the adaptive one. Like,
it's part a matter of timescales. So one of the things that came out of the work that we did on measuring causal control, causal specificity, was realizing that in a lot of the philosophical discussions of, which cause is the cause of this, right? People are very cavalier about what the variables are and what the data sets over those variables are. So if what you're talking about is, you've got to be clear about whether you're talking about over an evolutionary time scale where the variation in the gene is, different alleles, what the alleles are doing, or whether you're looking on a developmental time scale where that gene is a variable, but what you're actually measuring is a time series of whether it's switched on or switched off, whether it's being expressed. Of course, more adequately expression levels, but that would get really hard, right? So if you build a little baby model to think about this on a developmental time scale, your variables are, the variable on the cause side is the state of expression of the gene in what you postulate to be the controlling genome. If you're thinking on an evolutionary time scale, the variable you're talking about is which alleles are there, right? So these are, there's a lot of...
quickly?
Yeah.
I was thinking on the developmental scale.
Right.
Thomas, is it okay if I respond?
Sure,
yeah, okay.
No problem.
So I was thinking about like a toxin-antitoxin type meiotic drive system, where you have the toxin and the antitoxin, they might not be in the same cell. It's sort of like really early embryo or pre-embryo. But in that case, you'd want to say that the organism that has the toxin but doesn't have the antitoxin is causally in control, but it's sort of not adaptively in control. It's
right. Yeah. No, that's really good. That's actually really good. All I can say about that is that I need to go and think that through, because I mean, my thought is not that these are necessarily linked. It's that the structure of causal control is going to dictate who finds it easy to evolve phenotypes that serve their interests. So if a little sort of example, right? If you're dealing with, say, facultative sex determination in birds, my sort of way of thinking about this would lead me to think something like, because sex determination is a factor in the egg, right? So what sex ratio you're going to get is determined by which eggs you expose for fertilization. That that causal fact about the structure of development in that species ought to make a difference to how easy it is to get control of that variable so that sex ratio serves the female's reproductive interests rather than male's reproductive interests. So I'm sort of thinking that the structure of development is going to set the agenda for the evolution of whatever system you spit out at the other end. So what I'm saying, I think those two variables ought to end, those two things that you're measuring ought to sort of coincide. It's a mixture of a developmental and an evolutionary timescale. It's if causally all of the variables you can manipulate in order to change this variable are in my genome, it ought to be easy for me to play with those variables so as to get the effect variable to have a value that serves my evolutionary interests. So there's, I mean, think about it again in placenta, right? It's you know, it's the physical facts about placentas and how they're made, which are set the agenda for that evolutionary competition. Okay, over you know, so we got this sort of nice, you know, let's not kill each other arms race balance, if you accept Haig's perspective, right? And where that balance sits ought to reflect, as it were, the mechanistic possibilities in the arms race for the two competing parties. That's the thought. It's very undeveloped, and all I can say about the democratic drive case, I should go and think about that and see whether it makes any damn sense given the way I'm thinking about this.
Thank you. We now have a question by John Dupre.
Thank you. Thank you, Paul. That's really interesting, and I'm very sympathetic to the general project. I guess my biggest reservation is that I'm a little concerned by putting all this in quite such competitive terms, and I guess at least I wonder whether there's a kind of complementary story that you might talk about in a more cooperative way. And I haven't really got very much to say, but I've got just a few, well, 2 or 3 thoughts about this. I mean, one is that, of course, the mother and the fetus are parts of much larger processes, and particularly a process of a lineage, and they don't, you know, a lineage is also something that has to persist to be evolutionarily successful. And I guess within that context, I wonder a little bit about the extent to which your competitive picture, and by the way, I quite understand the argument that you've got to, you know, that enables you, that perspective enables you to take account of a lot of the scientific work. So I'm not, I mean, I think there's lots of interesting insights that you're getting from that perspective, but also they in a sense forced you to take this individualized perspective, in which there just are these two entities that have this range of interactions with one another. And I guess one of the things that seems to me valuable about the process ontology is that it doesn't force you to recognize this kind of countability of entities as something fundamental and immutable. And I guess it seems to me that the one or two entities, which I think maybe I read Elsa Lyons slightly differently, but you seem to read her as saying there are these two things, but then there's bits where there are bits that are parts of both of them. Whereas it seems to me that maybe there's more, that's less ontological and more pragmatic, that there are ways in which, so I'm not sure if that's right or not. But at any rate, it seems to me you can't, when you take your kind of gene-centered, who's in charge, who's in control, you're kind of forced to take a particular kind of counting of the entities, which in a sense maybe begs some questions. And I guess just a much smaller sort of concluding thought. I was just struck at the very end when you were talking about the kind of extended control of the mother by the offspring through microchimerism. I mean, it's sort of struck me, but clearly the fetus the fetus, the infant continues to, the mother is part of the infant's extended phenotype. I mean, she's feeding it, she's taking care of it, she's doing it, and there may be an effect through the microchimerism, but it seems so marginal. And I guess it's I wonder whether you're kind of getting drawn a little bit into
that stuff is wildly speculative, John. I agree. I mean, it was
but it's not just that it's wildly speculative. It's like it's a wildly speculative addition to something that's obviously the case and wouldn't really make much difference to something I would have thought that was obviously the case if you're more, you know, kind of in the context of a narrative that takes more seriously the cooperative aspects, the aspects where the processes intersect and then combine to do things together rather than compete to see who's doing them. So that was the
yeah. Again, you know, there's a thing about whether you're thinking about sort of evolutionary actors and lineages or whether you're thinking about individuals. I mean, my own interest in this came out of a project with some of the guys in our center looking at the evolution of preeclampsia. So why this, you know, why do we get preeclampsia? And, you know, so there's an enormous amount of cooperation in the same way that at the end of the process, you know, nobody wants to kill their mum and nobody wants to kill their baby. All right. These are really bad outcomes for every participant in the process. But on the other hand, you know, exactly how much you're going to feed me is something that we might have a significant disagreement about. Okay. So I mean, on ontogenetic level, you're looking at, as it were, you know, people implementing an agreement. But the source of that agreement reflects their capacity to negotiate the agreement over evolutionary time. So I think things look less competitive when you look at this on an ontogenetic timescale. Okay, but if you're trying to understand the emergence of the system over time, I mean, in the case of preeclampsia, it's really weird, right? It's very, you know, it's incredibly expensive. It's a disaster for all concerned. It's really common. It's a ubiquitous problem that is faced by women, right? And data is a bit dodgy on other species, but we assume there are problems in other species too, but you know, they don't come to the health system, so we don't have quite enough data on that. So you're trying to understand how we could have incurred this kind of massive ongoing cost. And, you know, a cooperative perspective doesn't give you that. In a cooperative perspective, we would have got rid of this crazy problem a long time ago. So on an evolutionary timescale, in that particular case, which is what got me into this, you know, the Hague stuff really pays off. But I mean, I share a lot of your concerns. I mean, one of the problems is if you want to actually develop stuff to the point of view where, you know, philosophical work is useful to scientists, you've really got to, you know, plug into, you know, the research, the science as it's currently being done. So, I mean, I would love to be able to do this stuff for, you know, developmental systems rather than just genomes. But when I do it for genomes, I can actually build baby models that make sense to my colleagues. Now, actually, I agree that that buys me a whole bunch of biases and historical inertia from the way those disciplines have developed. And all I can say about that is, you know, yep, that's a problem, and it's a real problem, and we should always remember it and wrestle with it. But I don't have much more to say than that. Yeah. Yeah.
Thank you very much. We have 1 minute, so I guess it's very, very limited for question and answer. Maybe someone has a very short question. Doesn't seem to be the case. So in that case, I think we should move forward.
I could just say happy to answer emails later on if people want to email me. Just do, please.
And I must say this is very likely that people send you emails, because it seems that this is what happened last time, and also with the YouTube videos, I think you might get other emails, which is also a cool thing with these remote workshops. So it's now time to listen to Thomas Bosch after indeed uploading again Paul for this talk. So Thomas Bosch is a professor of general zoology at Kiel University in Germany. He's the spokesperson for the DFG funded collaborative research center called Origin and Function of Meta-Organisms. And indeed, Thomas is one of the main promoters of the concept of meta-organism, co-promoted, in fact, with Margaret McFall-Nye. You saw her last time. You will see her next time if you can connect. Thomas has been doing in the last 20 years or so fantastic physiological work on Hydra, especially, not only on Hydra, but Hydra is his main model organism. And he has worked on Hydra to better understand host microbiota interactions, immunity, the nervous system, but also aging, regeneration. So very important topics in biology, and also, of course, in for philosophers of biology. And Thomas today is going to talk about nature's oldest neurons interact with bacteria. So Thomas, thank you very much for accepting the invitation, and the floor is yours.
Thank you very much, Thomas. Thank you for the invitation. It's a great pleasure and honor to talk to this audience. It's also we have now to switch gears after Paul's philosophical thoughts, but I think we stay somehow in the idea that organisms are multi-organismic associations. In Paul's talk, maybe overlapping individuals, but which form one entity, which we call the holobiont or the meta-organism. And today, I'm generally interested in how does this work in terms of how do the interactions work between the different individuals within one holobiont or within a group of holobionts, which we then call a meta-community. And today's, I will just focus on recent findings on the nervous system. And what I'm going to do is I will briefly mention the ideas about the origin of the nervous system, and I'm introducing my model organism, Hydra. Then I will tell you that neurons talk to bacteria. I will tell you that bacteria activate neurons. We have identified recently the prototypical pacemaker cells, which are in your body as in any other animal body. And I will show you the molecular identification. And I will end up with giving you a view of the molecular architecture of an ancient nervous system, which very clearly did evolve for interacting and with the environment, in particular with the microbial environment. When we think about the origin of the nervous system, there's a few groups which are interesting in terms of that they have somehow maybe the most oldest nervous systems. And this is two groups, the ctenophores and the cnidarians. I will focus on the cnidarians, which are a sister group to all of the rest of the animals, the bilateria and the bona fide neurons and nerve net evolved just in the common ancestor of the cnidarians and the bilateria. For that reason, if you want to understand something about ancient nervous systems, cnidarians are a very good place to look at. The cnidarian I'm working on for all my life is freshwater polyp. It's a very simple model system. It has a single body axis. It has only two cell layers, an ectoderm and an endoderm, made up of different stem cells. Intermingled there are neuronal cells. We have understood the whole cell biology, differentiation pathways. We have the genomes. We have developed all the genetic tools to manipulate every single cell in that organism. We still don't understand how it really works, but I think we may understand it better than in many other model systems. Very simple Bauplan, diploplastic, no mesoderm. That's important. And I think of Thomas Pradeux in Bordeaux, and he thinks about immunity. The immune system is a mesodermal derivate. Here we have no mesoderm. The animals are extremely well protected, but they use the immune system to entertain and to regulate the holobiont. In the absence of a mesoderm, the question is what type of cells are doing that? And you will guess, because the title's talk is that nerve system is a major part of the immune system, and that's a shared commonality with all other animals. Multifunctional cell types, and there is no specific immune cells. All the cell types are multifunctional. The important point, what I want to tell you, is that actually the nervous system, with that reasoning, evolved prior to the immune system, prior to the mesodermal immune system, which we are used to think about, the adaptive immune system, which is clearly a mesodermal derivative. So this is only ectoderm and endoderm, and the nerve cells are ectodermal derivatives. In addition, these animals have no centralization, no cephalization. That is just a nerve net which is surrounding a gastric cavity, and that is nothing else but what mammalian people call the enteric nervous system. You have billions of neurons in two of your nerve systems, in your intestinal nervous system as well as in your central nervous system. These animals have only the enteric system, and they are much older. So it's reasonable to think that the enteric nervous system evolved first. The nervous system is well described since hundreds of years with different technologies. Not this one. This is developed in our lab. It's transgenic neurons expressing GFP. And you can see it's a nerve net. Neurons are in direct contact to each other and intermingled in the muscular epithelium. There are two cell types in the neurons. These are ganglion cells and sensory cells. It's a nerve net. But this is known for a long time, chemically very complex. So there are sensory cells around the opening, the mouth opening, and then there's a ganglion nerve net. The astonishing fact in these animals is since they grow asexually by budding, the nerve net is continuously building up. If you want, non-senescent forever. And it can do that because there is a powerful population of stem cells, and these stem cells give rise to many different cells, including neurons. So in adult animals, we have a continuous neurogenesis, and how you can build a specific nerve net, which is in every single individual identical, in an animal which continuously grows and rebuilds the nerve net every single day, is still very much enigmatic and is of high interest also in my group. Here I focus on an astonishing observation which we did some years ago. This is a beautiful picture where you see in blue the nuclei and in green the green fluorescent expressing neurons. But you see, if you look carefully on the right spot here, you see some red spots, and these red spots are bacteria. And we have observed many years ago that there is a stable symbiotic community outside the ectoderm in a glycocalyx, species-specific, absolutely essential. If you remove it, the animals are not functioning anymore. And here we are obviously in very close neighborhood to the neurons. If you go by electron microscopy, you see a variety of different microbes on the surface of the animals. And schematically, you see what we could observe in many different cases and with different technologies. Sensory and ganglion neurons go with their protrusions directly in the glycocalyx in your body, that would be the mucosa, where the microbes are living, and they obviously have at least direct contact with the microbes. The question is, what are they doing there? So we thought that, how interesting. We are interested in how a holobiont functions. But this holobiont has invented a new organ. This is the nervous system. So question is, how does the nervous system, a newly evolved organ, is getting integrated into a holobiont? Is it taking part in the communication with the microbes? What do the microbes do with this newly evolved organ nervous system? And I give you 4 small chapters to tell you what we learned in very summary, and you can read that in the papers, the details if you're interested in. The first message is neurons talk to the bacteria. And the evidence came because we found a neuronal derived antimicrobial peptide, a gene product, which is produced in neurons, in very specifically in neurons. And you see here ganglion cells, and the ganglion cells go with the protrusion through the epithelium in the outside world where the bacteria are living. And when we do here is in a higher magnification. This is the neuronal nucleus. The green spots are the vesicles filled with this peptide. The nuclei release, the cells release that peptide. And if we do genetic and knock out that gene, then we can find that we have a disturbance in the outside microbiome, in the symbionts, in their abundance, in their composition, and in their spatial organization. So it seems that a neuronal derived gene product has direct influence on the normal distribution of the microbiome. Neurons talk to microbes. Do microbes talk to neurons? Question mark. Here we go and look a little bit in nature, and what we realize it, sorry, I have to activate the movies because they're so nice. If you go in nature, you find, and this is still a mouse, but this is you now. This is your small intestine, and these are this is another mouse intestine, and this is of course a coral. I call it the pulse of life, because all these animals can do spontaneous contractions of their bodies. There is kilograms of papers who are trying to understand why are they doing this. When we first found that, we started to ask the question, how are they doing this? And we got intrigued by that, because if you stop and if you limit these contractions, animals get sick. And you get sick, that is your intestine, your small intestine, and you have constipation, and you have all kind of trouble if that is not functioning normally. So the question is, how does that work? And so you use the model system. I had a fantastic graduate student, Andrea Muggillo. She's now a postdoc in a nearby Max Planck Institute. And we investigated that systematically by bringing cohorts of animals under examination desks, these photo cameras and these computer systems, so we can measure and quantify the contractions. And so we could measure the amplitude of the contraction. We could measure the regularity of the contraction. We could measure the time of the contractions. All of that very, very nicely. And the absolute, for me, unexpected, surprising, and astonishing observation was that when we removed all of the microbes from the animals, the contraction frequency dropped drastically, and not shown here, also the periodicity of the contractions was greatly changed. That's what causes your pain. If you have constipation, you have, because the rhythmicity of these pulses, the periodicity is disturbed. So this is not shown here, but this is also the case here, but you can see there's a drastic reduction. And sure enough, if we add back the bacteria, we can rescue that. We can even rescue that if we give back bacterial extract. So that was exciting, because it told us that in this evolutionary old and ubiquitous behavior of spontaneous body contractions, microbes play a role. And very clear, these body contractions have a nervous background. Because in nerve-free animals, we have a mutant where you can remove all the nerve cells. of course, they cannot do this body contraction. So microbes are involved in these body contractions. And the model at that time was, we have a complex microbiome, which is sitting outside, and it produces something, and this something goes to a not yet identified receptor on a neuron, and this neuron then activates the epithelial muscle cells, and that then leads to the contraction. I stress here, because it's a general rule in the host microbe interaction field, that it's not a single bacteria species which does the job. You need a complex community, and the trouble and the disaster of our days today is that we systematically ruin the complexity of our microbiomes. So then when you have reduced the complexity, they are not able anymore to produce these molecules and then to activate them. That may be one of the reasons why inflammatory bowel disease and constipation is increasing so dramatically in the modern time. So that's the model at that time. And the question of, we published that, and the question, of course, was, who is the receptor? And how, which type of neurons are involved, and how does that work? Can we go deeper in the genetics and in the understanding of that process? Here, an unexpected twist in the story came because, as a simple-minded general zoologist working for 40 years on Hydra, you cannot solve the problem. So I met a wonderful group of people, and these are human doctors or human biologists, and they are interested in this phenomenon of diseases associated with spontaneous body contractions. And one of the diseases which my colleagues here in my institution are interested in is irritable bowel syndrome, or IBS. That is associated or accompanied by dysmotility. It's increasing in the last 70 years. It correlates with a disturbed microbiome, but causality in humans is not known. And I started a very fruitful collaboration with my colleague Mauro D'Amato. Mauro is a world expert on IBS, and he had a fantastic experimental platform because he sequenced 11,000 Swedish twins, and to get to the molecular signature of IBS patients. And among these there were 500 IBS cases and 5,000 asymptomatic controls, and he had a long table of genes involved in that to understand the molecular biology of intestinal motility disorders in humans. In humans, you cannot go really for causality. If you want to go for causality, you need animal models, which then are informative to tell human biologists and medical doctors, well, that's an interesting way to go. And that's exactly what we did. And there is one more bizarre observation. And the bizarre observation is that fecal microbial transplantation helps in many of these dysmotility cases. Now, my colleagues and others have asked what the hell is in that soup here, which is then getting transplanted from a so-called healthy donor into a patient, and we still don't know yet the atomic composition, but we know it's nothing but microbial products, components, maybe phages, but mostly microbial metabolites. If you know that, then you can think, well, can microbes help? And then you think back of our experiments in Hydra. Nothing makes sense, but in the light of evolution, of course. And so we started with our medical informed approach, a huge effort, and started from the human GWAS study on IBS patients and ended up with a huge genomic effort in Hydra. These two absolutely fantastic colleagues, Alexander Klimovic, he's a senior postdoc in my lab, and a very young student who entered last year. He's now master student and PhD student, Christoph Dietz. And what we did is we used, in close collaboration with Mauro, the human data, went to Hydra, but then we not only got the genes, but we did then functional genetic experiments with the human identified genes in Hydra, and then we went back to the mouse. It's a long way. It's published in a few weeks in PNAS, and so we, of course, identified some of these signatures in humans are ion channel proteins. I mentioned here 3, SCN5, ANO34, and TRPM5. And all of them are highly conserved, and we found this in the Hydra genomes and transcriptomes, and very closely related to the human ion channels. So the nerve cells responsible for that microbial contact seem to express ion channels, which makes a lot of sense and tells us maybe we are already at the pacemaker population. We then identified which type of cell this is, and what you see here is this is the ANO1 expressing cells, and beautiful, limited number of ANO1 and also SCN5 and TRPM5 around the head, and we know from beautiful old papers, more than 50 years old, that this is the pacemaker region. And just to give you that the research I'm doing is also beautiful. We continuously, or my colleagues continuously produce pictures which are just outstanding. What you see here is the few, about 20 pacemaker cells expressing these 3 ion channels. And if you remove these guys, you remove the spontaneous contractibility. And we showed that by using inhibitors against these ion channels. And if you inhibit these ion channels, you reduce the contraction frequency dramatically in all these cases. So this old creature is not only responding to microbial products, which come from a complex microbiome, but also the receiving cells of these microbial products seem to be a small group of neurons, which we classify as pacemaker cells, which we found because a disease phenotype in humans, and which are characterized by expressing a certain highly conserved group of ion channel proteins. So in continuation of the model, it's not only that the complex microbiome produces something, but we also now have identified the receiver, which is the host neurons, and the host neurons are the signature are these ion channels, and here are chemical interference molecules where you can block them, and then you block the spontaneous contraction. I should add that you only block the spontaneous contraction. You can squeeze the animal and it will contract. It can somersault, it can move, it can behave. Only this spontaneous contraction is inhibited. Very specific. This is due to this specific type of cells. So part 1 is that it seems that the prototypical pacemaker cell is characterized by ion channels, also by gap junction proteins, by the way, because these neurons make networks with other neurons, and they are in direct contact with the gap junction. We also have shown that. And the approach to that was that we learned from our medical biologists because they are interested in an important disease. If you are so far, then you can say, well, we have all the technology now, and it seems that we can tell the neurobiologists around the world something about how it really functions. So we may go one step further, and we may try to now get to a more complete picture of the molecular architecture of a complete nervous system, but this nervous system is one of the oldest nervous systems in nature. And that's what we did. And we did that by single cell transcriptomics. we had genetic markers in every single neuron. The animal has 3,000 neurons. How wonderful if you think that you and I have billions of neurons. We never can understand that. But these guys have 3,000 neurons. We know every single neuron by first name. We have integrated genetically in every single neuron fluorescent markers, green fluorescent, red fluorescent. We are working on spaghetti hydra, which are expressing all kinds of different colors in their different neurons to get to the complete picture, probably the first picture ever on a complete nervous system. And what Alexander and his colleagues identified is, so we did single cell sorting based on the fluorescence markers, and we distinguished from stem cells and we distinguished from other cell types, and we found 7 distinct neuronal subpopulations identified by their similar gene expression pattern. And what you can see is they all are very distinct. So these simple, and please never say primitive, this simple nervous system is enormously complex. It is consisting of many different subpopulations. They all talk to each other. And one of the populations must be the pacemaker population. And that's what we are going to. We have developed all the technology. You see here now live imaging of calcium expressing a calcium reporter construct expressing neurons. So when the neuron is activated in the live animal, it's flashing. And we can also do that in a more complex situation where we see now every single neuron live in action. So if the microbe comes, we can now identify which type of neuron is firing at what time point. With this knowledge, we go back and now try to construct this nerve net. And the single cell profiling, of course, showed us, first of all the conserved and typical genes, which are immune for neurons, receptors for neurotransmitters, transcription factors, neuropeptides. Some of them were known for quite a while. We and others have identified that by completely independent approaches. So that confirmed that these neurons are highly conserved, maybe the prototype, and many genes which are also in our nervous system is already there in this old nervous system. That was something to be expected, not very surprising. The unexpected and really, for me, very informative and mind-changing observation came when we realized that the majority of transcripts in these neurons are immunogenes. About 70% of the transcriptome of neurons you can classify in immunocategories. This is now the 7 groups of the neurons. And this is, for example, the genes which you could categorize in the toll-like receptor pathway, the MID88. These are genes in the NACH domain. These are C-type lectins. These are antimicrobial peptides. These are Causal domain peptides. We have identified many other peptides, and we find them now in neurons. Immune receptors and pathways are predominantly expressed in these neuronal cells. It's getting even worse because, of course, if you do such a quantitative approach, you discover many genes which you think, well, they make no sense because they are not conserved. I cannot find them in any blast program. And so you call them orphan genes. We call them taxonomically restricted genes. You may call them novel genes, whatever. These genes are highly abundant in neuronal populations compared to stem cells. If you see here, this is the number of the percentage of TRGs in stem cells. And these are terminally differentiated cells in hematocytes, and here the neurons about 70% of the transcriptome of neurons is not conserved. God help, what are they doing? Now, many years ago, I think in 2009 or something, we stumbled on that genes. Nobody was really interested in that. We placed a hypothesis paper in Trends in Genetics after a PLOS biology paper with original information, where we proposed that these genes must be important because they are present in every single organism, these non-conserved genes. And here, this is from our old 2009 paper. In blue is the percentage of genes which have no blast hit in all kinds of different organisms. And you can add humans and anything, and people ignore that because you cannot make much sense out of these genes. But every single species has species or taxon-specific genes. We strongly believe that these genes are very important and they contribute to the species-specific phenotype. But finding the function is very difficult because you have to do literally genetic with every single gene. You have to knock it out, you have to overexpress it, and that's very laborious. We did that with one or two, but now we have a much more elegant way. And in addition to Mauro D'Amato, this effort was joined by a fantastic group at UCLA, Edward Wong's group, who are doing machine learning. And when I talked to Gerard, he said, well, just send me your novel genes, and I feed my computers with your novel genes, and maybe we can make sense out of them. And we feed the computer programs with some predictions. For example, so this is the amino acid sequence of a novel gene. And we ask, can these novel genes be membrane transfusing, have that membrane transfusing features, which some of the antimicrobial peptides have, for example. So we asked this kind of questions, and Gerard and James programmed their machine learning computers, and we sent them our sequences to UCLA. And sure enough, we got super interesting ideas. There is a sequence of amino acids from a novel gene. And when we look at that, and we are a little trained on looking at antimicrobial peptides, we immediately saw, my goodness, that could be an antimicrobial peptide from a new gene. So then we did the functional test, and sure enough, this is a novel gene predicted by the machine learning program from Gerard Wong and Jane that's interacting with microbes. And we did that in MIC assays and in other direct assays, and we sure enough found that these novel genes, many of them, also contribute to the communication with the microbes. They do communicate, and that may sound a bit funny, but they communicate by killing. But the communication is that they maintain the healthy microbiome, the stable composition of the microbes by secreting a cocktail of antimicrobial peptides, and each member of this cocktail has different specificities, and by that you maintain a stable microbiome. So in addition to the conserved genes, and 70% of the conserved genes are already immunogenes, neurons have expressed many other genes which also are immunocompetent, which means neurons are immune cells in these organisms. So to conclude, we think organisms are multi-organismic associations. We call them meta-organisms. Here's the host, here are the microbes, we have viruses, we have symbionts, they live in a given environment, and the communication between the host and the bacteria is done to a great deal by neurons The neurons secrete molecules, including antimicrobial peptides, which then take care that the symbiotic microbiome, which is outside the animals and are glycocalyx, is of a given structure. And if you disturb that microbiome, the animals get very sick and very different. It's the same as if you take antibiotics and your microbiome is getting sick and different. The story comes to an end at the moment, and that's the final figure of the publication, which comes out in a few weeks. Because of the collaboration with our colleagues from the mouse field and the C. elegans people, we could show that this is the prototypical pacemaker cell. It evolved as a neuron, but in addition to the very well-known neuronal genes and the neuronal pathways, these neurons from the very beginning of evolution were full of immunocompetence. The ion channels, the antimicrobial peptides, the conserved and non-conserved genes. This is a very distinct set of neurons which are responsible for one specific task, which is the spontaneous body contractions. The same is true in C. elegans. We have identified these genes also in a certain group of pacemaker cells in C. elegans, and the same is true in the mouse, where we have these interstitial cells of Causal, which are the pacemaker cells. These are neuron-like cells, and in our body, they function as they do in the Hydra. Identification of a prototypical pacemaker cell, which from the very beginning of evolution really was involved in communicating with the microbiome. So I come to the end, and I just want to finish this morning, or in Hawaii, late night, evening, by just repeating once more that neurons, not only in Hydra, but in the animal kingdom, should be considered as immunocells, that they express distinct classes of neuropeptides and cytokines which regulate the microbial community. That happened at a time when there was no mesoderm. That happened at a time when there was no adaptive immune system. So we think this is part of the origin of the immune system, and one has to look in the nervous system to find that. From the beginning of animal evolution, neurons received signals from microbes, and they interacted with microbes by secreting antimicrobial. So in addition to sensory input and motor output, what nerve systems, if you look in a textbook, usually do, I suggest that we have to add the nervous system has an intrinsic property of interacting with the microbes, and it has multiple functions in host microbe interactions. And with that, I thank my colleagues here in the team, Christoph and Alex. Gabriel Kruppi was a master student contributing. Andrea Mogillo did beautiful early work. Mauro D'Amato, now in Australia. Igor Adameko, I should never forget, he did the mouse work, and Gerard and Jane, who did the machine learning. I run a center here in my institution, Origin and Function of Microorganisms. I'm sitting right now here outside the sunshine, but the campus is empty, but I'm very happy to have an audience and to see you. Thank you very much for your attention.
Thank you very much, Thomas. That was great. Let me, switch to the gallery view. So I think if anyone had any doubts that Hydra was a great model, I think everyone now is on board and everyone is convinced. So really great work indeed. We have, let me see, around 13 minutes for questions. So does anyone have a question right now? Maybe you need a little bit of time to digest, so to speak, all these elements. I have one question, Thomas. Antimicrobial peptides are used by plants to communicate with microbes to a large extent, presumably without a nervous system. So are we here talking about different ways of dealing with microbes, which in animals would go through the nervous system and then the adaptive immune system, but would have taken a different route in plants?
Thank you. It's an interesting question, of course, and there are two answers. Plants are different from animals, very clear, and plants have no nervous system. But the way how to talk to microbes is conceptually quite similar between plants and animals, even in immune pathways. And if you remember, for example, the NLRs, the knot-like receptors, which are intracellular receptors, and in animals also present, but these are the main receivers of microbial signals in the plants. So yes, in the absence of neurons, there is a hidden commonality in the biochemical pathways to deal with microbes, but of course, when there are no neurons, the neurons cannot play a role.
So just a quick follow-up, because I don't see any other questions at the moment. So it means that, okay, I see questions now. So I get back to this if we have time. So Anna.
Hi, thank you very much for this wonderful talk. I wanted to make sure, so these ion channels, you think they are the receptor that interacts, or you think there's another receptor that interacts with the microbial products? Or you think these ion channels are directly binding to?
That's an excellent question. We have not yet shown that because we have not yet identified the microbial product, which is, we are trying, as you can believe me now, for 4 years, and which needs state-of-the-art mass spectroscopy. And since we are dealing with extracts from number of bacteria and not from a single species, it's getting really complex, but we are on the way. But unless we have not identified the ligand, it's hard to say how is the activation of the receptor. And you are completely right, it can be direct. We rather think that these ion channels are in the second row and that they are activated by a not yet identified receptor which binds to the ligand. Very good question. You have to unmute.
Yeah, turn on your.
If I may ask a second question. So you've shown that this system was conserved from C. elegans to Hydra to Max. Have you heard of insects? I'm working on insects.
That's a good question. There is a reason why we didn't look in Drosophila. Probably Drosophila is so complex. I don't know. I would assume if you are, I don't know your background, but if you are in the neurobiology of Drosophila, there may be some cells which have this pacemaker activity in the gut. And I bet that if you would identify them, that you will find this ion channel signature there as well. And I'm very much convinced that this is a highly conserved signature. Drosophila is very weird in many aspects, as you know. It has lost some of the genes which the rest of the animal kingdom has. So if you do not find them, too bad for you, but they are conserved.
For sure. Thank you.
Other questions? I have another question. Oh yeah, Gregor has a question.
Thanks a lot for the fascinating talk. That was very interesting indeed. So my question was about what the immune system regulates in terms of you pointed out the composition and the spatial localization of the microbial community. And I was wondering if there were other aspects you would take into account that the neurons or other immune functionings, different cell types would rather interact not with the localization composition, but rather in inducing different gene expression, modifying not the community or localization aspect, but rather like fine-tuning the activity of those microbes that are already there rather than changing which microbes are there.
Thank you very much, Gregor. I'm not sure if I really get the point, but I think, and what I have to say is, I guess, and I have to paint a bit more complex picture. Meta-organism consists of about, Hydra, consists of about 100,000 Hydra cells. They are most of them are multifunctional, and epithelial cells, for example, also express antimicrobial peptides, not only the neurons. How epithelial cells talk to neurons, in any way, how a cell lineage talks to another lineage, is not known in any organism, and we are interested in that. The immunity is in two ways. Immunity is maintain a healthy microbiome outside, although we do not know yet what that really is. But the most important part of the immune system is indeed the microbiome. And we know that because if you remove the microbiome, animals get immediately attacked by fungi, for example, and they are completely immunocompromised. So you do a good job if you maintain this symbiotic co-evolved microbes, and you do that by a super sophisticated stoichiometry of molecules which maintain the composition and the regional organization, etc., of this microbiome. The microbiome's part of the immune function is, to a great deal, to prevent intruders to colonize the tissue. We call that colonization resistance. So if everything is fine in your gut, in the best case, or in your skin, in the best case, all the niches are occupied where any other guy can settle. That's niche exclusion, and so then nothing can happen. The trouble comes if there is a real bad guy who manages to get through your microbiome and then go into the host cells. And then it's a pathogen, and then you have to activate some direct aggressive means to try to kill that pathogen. But the predominant role is maintain the healthy, beneficial microbiome, and they will do an important job to preventing any environmental microbe to settle.
Thanks a lot.
Thank you. So there's a question by Lynn. Lynn, it seems, okay, Lynn, you're here?
Yes, I'm here. I'm sorry that I don't have video access today, and it's good to see and hear from you again, Thomas. So I have a question about, a very basic question about the relation between, in Hydra, spontaneous contractions and whole organism behavior, especially behavior that can be considered purposeful or related to information processing.
Okay, I have also to think how to answer that short. We are very much interested in now, as you can see, to understand the relation between microbes and neurons. And Hydra has not only the beauty of having only 3,000 neurons, which we can study every single one, but it also has a behavior. And all of that was forgotten. And you have to read old papers to learn new things, as my peer Pierre Tardin also told me. And so I picked up these papers on Hydra behavior, and there's, as you say, spontaneous contractions. They go and they're spontaneous. And we think that this pacemaker population is responsible for that. But there is also what I would call, Lynn, I would call reactive behavior. And that is, you give a stimulus, and then the animals behave in a predictable, reflex type of manner. And we have selected, studied that research 2 years ago. It's not yet far away from getting published, but we study a so-called feeding behavior, which is a reflex but it's reactive. It needs a stimulus, and then it goes and does the behavior performance. And again, I can tell you that germ-free animals they have parts of that behavior, but they are in trouble by making the full behavior. As germ-free mice have trouble to behave normally, germ-free Hydra have trouble to behave normally in the absence of microbes. And here we are in the ancestral communication between neurons and microbes in terms of behavior. And we are trying, and this is Christoph's PhD thesis, we are trying to understand that at the molecular level as deep as we can do. So yes, we left now the spontaneous contraction, and we go now to a reactive behavior, which is far more complex and to understand the neuron microbe interaction.
Thank you. Thank you. That's really exciting.
Thank you very much. We have 2 minutes. Tyler, you had a question. I don't know if you still have a question.
Sure, it's a simple question. I wanted to know basic anatomy of the Hydra I'm not familiar with, but the outer layer is the glycocalyx.
So the Hydra host is diploblastic. These animals are radially asymmetric and have only an endoderm and an ectoderm.
Okay.
And people focus on that ectoderm and endoderm for the last, since 1743, invented by Abraham Tremblay. But what modern sequence technology told us that the ectodermal cells secrete a sugar layer outside. Right. And this sugar layer is what you secrete with your intestinal cells in your intestine, and you call that, or the doctor calls it, a mucosa. And in this extracellular layer, the microbes are living.
So I just wanted to know if the effect of the neurons on the distribution of microbes was mediated by some effect on the glycocalyx. Does it change the glycocalyx and that attracts the microbes?
Yeah, so the answer is very clear, no. So that's a direct effect from organism to organism, from host cell to microbes. You may say from individual to individual. However, in the long-term absence of microbes, the glycocalyx structure and anatomy changes, and it's getting thicker, as if the cells would know we have to make a better habitat that microbes can live there. So in the long run, germ-free animals have a changed glycocalyx because that's a purely host-derived structure. But the effects I'm talking about is direct from one organism to another one, and not via changing the glycocalyx.
Thank you.
Thank you very much. We're perfectly on time. So I just want to say, as you just saw with these two talks, there are very different ways of addressing the question of biological individuality, but this is a very important question first. And second, as Thomas said, I think the question of overlapping is very important. We will also see in a minute or a few minutes that there's also a very important question in terms of, are we talking about an individual, or are we talking about a group, or maybe about both? And what does it mean to say something of that sort? With this, and I'm applauding again for your talk, Thomas. That was really great. Thank you so much. I think we will resume in 5 minutes to be on time. So it's 11 here in Bordeaux. I suggest you cut both your microphone and your camera, because I'm still going to record the meeting for the sake of simplicity. So please, yeah, turn off everything. Thank you very much to our two first speakers. And we start again in 5 minutes with Samir Okasha's talk. Thank you all, and see you in 5 minutes. I see that at least some of you are back. It's of course pretty essential that Samir be with us. Samir, are you here? Oh, here's Samir. Okay, I think Samir, we can see you and we can hear you.
Yeah, I just got to share my screen and then I'm ready.
Okay, very good. So just a few words of introduction to state the obvious, because you all know Samir Kasha. Samir is professor of philosophy of science at the Department of Philosophy at the University of Bristol in the UK. Clearly also a central figure in philosophy of biology. Samir is a specialist of foundational and conceptual issues in evolutionary biology. He's a world-recognized specialist of levels of selection in evolution and related issues regarding biological individuality. He's the author of the book Evolution and the Levels of Selection, which was awarded the 2009 McIntosh Prize. And interestingly, this is one of the very few books you can see, books of philosophy, you can see on the shelves of basically all evolutionary interested biologists in the world. So, I mean, this is a very interesting thing that Samir achieved of being read and discussed and commented by many evolutionary biologists in the world. Samir is also the author of a book called Agents and Goals in Evolution, published by Oxford University Press in 2018, and also more recently, a very short introduction to philosophy of biology, also at Oxford University Press. So that was, I think, last year. It's a real pleasure to have you with us, Samir. And your talk today is on the very idea of biological individuality. So the floor is yours, Samir. Do you need more time?
No, sorry, I'm just rearranging so the screen wasn't so dark, but that's fine. Thank you very much, Thomas. So what I want to talk today about is the notion of biological individuality, and I want to try and articulate a disquiet that I've long had in regard to this debate, which hasn't been easy for me to make clear. So the themes of the talk are as follows. Firstly, the debate in both philosophy and biology over biological individuality. A couple of words about the origins of the debate. Most of the time I'll spend offering a diagnosis of where the debate goes wrong, but before that I'll try to persuade you that something has gone a little bit wrong in this debate, and then I'll trace some of the implications of that diagnosis. So this is a somewhat unusual talk, for me at least, in that I'm trying to apply a very traditional philosophical methodology using what's often called conceptual or linguistic analysis to a problem in the philosophy of science and indeed in science itself, which is not something I normally do and not something I would recommend as a general methodology, but in this case, I think it's called for. Okay, so there's an old biological discussion going back, at least to the 19th century, probably earlier, regarding the notion of individuality expressed using that word. So Julian Huxley, John Hopper, Leo Buss, Stephen Jay Gould, I pick out as scientists who've made a lot of play with this notion. The discussion appeared to enter philosophical consciousness in the 1970s, thanks to the work of David Hull. And it was related to debates going on then about so-called unitive selection, and has led to a burgeoning literature which has sort of seems to have resurfaced in the last 10 to 15 years or so, and has produced some extremely interesting work at the interface of philosophy of science and biology. And the current debate, I think, is often set up as follows. In the first couple of paragraphs of a typical paper on this topic, people will often set it up in this way. They'll say, well, look, the concept of a biological individual is both central to the practice of biology, past and present, so it's a central organizing concept in the science, and secondly, is much more problematic than it seems. And the reason given for two is that the paradigm biological individual is said to be a metazoan organism, multicelled animal, and the metazoa are characterized by a number of features. And these features include genetic homogeneity, nearly enough, genetic uniqueness, nearly enough, having contiguous parts, that's say parts that are physically connected to one another, having pretty clear spatial and temporal boundaries. It's fairly clear when the organism starts, when it stops, what its spatial extent is, having a life cycle with a bottleneck, with a single cell bottleneck, being functionally integrated and exhibiting physiological unity. And authors then go on to point out that there are very many biological entities that satisfy only some of those criteria or only partially satisfy them, in that satisfaction of all of those criteria is a matter of degree. Someone thinks of clonal plants, colonial organisms such as marine invertebrates, bacterial biofilms, social insect colonies, and entities like symbiotic associations, symbiotic unions, and here the observation that a typical metazoan is itself, in a sense, a symbiotic union is often made, and then entities that are sort of on the borderline of being real organisms such as viruses. So for all these reasons, it's argued that the notion of an individual, of a biological individual, is problematic, and hence there's a need for some sort of clarification. Now that all seems very well, and I'm on board with much of that, but I endorse the opinion that Thomas Pradieu voiced in 2016, where Thomas wrote, "There's considerable confusion over the definition of the notion of a biological individual and related terms." I think that observation is exactly right, and I think it reflects a sort of interesting disconnect between the literature in philosophy of biology since the 1970s and the broader literature in analytic philosophy. Now, it's not my aim to achieve a rapprochement for its own sake between these two literatures, but I do think that on occasion, some of the clarity that analytic philosophers pride themselves on, allegedly achieve, is useful for organizing debates in philosophy of biology, and this, I think, is a case in point. That disconnect has historical roots, but I won't go into that for now. So what's gone wrong exactly? Well, the diagnosis that I want to offer is as follows. Sorry, I just need to move my screen around a little. The diagnosis I want to offer is as follows, that the debate over biological individuality, as usually posed in both the philosophy and the biology literatures, presupposes that the expression biological individual is a sortal term. I'll say more about what that means in a moment. That's a term from the philosophy of language. But it isn't. And so therefore, the debate is sort of premised on a kind of category mistake or logical error, which is not merely a terminological matter, but I think has pervasive consequences for the way that this debate has proceeded. So that's what I'm going to try and argue. But I'll start by trying to motivate my sort of inchoate conviction that something has gone wrong here by offering you 3 brief puzzles about the debate on biological individuality. Firstly, what's so special about biology? Secondly, Hull's concerns versus contemporary ones. And thirdly, the dichotomy between biological individual and organism. Okay, so puzzle number 1. Now ask yourself this. I mean, why is it that we find this extensive literature on biological individuality, but no corresponding literature on chemical individuality, astronomical individuality, or neurological individuality. Why is it that biology, of all the so-called special sciences, is the only one that seems to have this proprietary literature? Why not? Well, I mean, I take it that whatever answer you give to that, it can't be that chemistry, astronomy, and neuroscience do not have individual objects in their ontologies. That seems to be clearly not true. On the contrary, we can sensibly talk about an individual molecule. You know, if you were a chemist, you might study one, an individual star, or an individual neuron. It seems to me unproblematic, since you can count molecules, stars, and neurons, to talk about an individual one of them. Second puzzle, Hull's work, as I said, played a formative role in the philosophical debates over biological individuality. But in fact, as is well known, Hull's main purpose was to argue that species are biological individuals, the so-called species are individuals thesis. But it's interesting to note that in the current debate on individuality, species don't feature at all, in that they're just not the sort of thing under discussion. Rather, the things under discussion are the things I mentioned before, you know, clonal plants, symbiotic unions, and so on. Species are just not in the picture in that debate. How then does Hull's concept of individuality relate to the concept at work in the current debates? That's puzzle number 2. Puzzle number 3, individual or organism. There's been explicit discussion in this literature of whether we should use the expression biological individual or organism. And many people treat this as a merely terminological matter, although some people have argued that organism is preferable, an opinion I'll endorse in a minute. But it's odd to think that this could be just a terminological issue. And indeed, this contrast to me is odd, because as we'll see, the expressions are quite different logically and semantically, for reasons I'll give. That's puzzle number 3. Okay, so what I want to do now is step back and do some sort of very basic philosophy on this debate, and just to sort of ask the question, what's going on? What's this all about? Let's focus on the word individual. Well, as used in ordinary vernacular English, it can be either an adjective or a substantive. So I might say, an individual chopstick is useless. You need a pair of them. It's the adjectival use. Or I might say, police are hunting an armed individual. Substantival use. Now notice that in one, individual modifies a count noun, chopstick. And notice that in 2, individual is really shorthand for person. So what we really mean is that police are hunting an armed person. So you see then that, at least in the vernacular use, the term individual typically, and I would say always, refers to an individual of a particular sort, though that sort may be only implicit. You have to glean it from context, as in 2. What about the use of the term individual in philosophy? Well, this is more complicated. Here I refer to Anglo-American or analytic philosophy, so-called. I think we can distinguish 3 senses. Firstly, what I call the logical subject sense. Secondly, what I call the unitary entity sense. And thirdly, what I call the sortally restricted sense. So these are my own terms, my own labels. I've invented at least 2 and 3. The logical subject sense. So in this sense of the word, an individual is simply anything that exists. So anything that exists is an individual. Or other authors will say an individual is anything that can be referred to with a singular term or that can be made the subject of a true predication. So for example, we find Bertrand Russell in famous 1903 paper explicitly saying that he intends to use the words term, unit, individual, and entity interchangeably. Russell then continues by way of giving examples of his use of these synonymous terms: a man, a moment, a number, a class, a relation, a chimera, or anything else that can be mentioned is sure to be a term. That's a very inclusive use of the word individual. Also endorsed by the philosopher Peter Strawson in his book Individuals from the 1950s. Secondly, a different sense, the unitary entity sense, endorsed by many modern metaphysicians. For example, E.J. Lowe, in his very carefully worked out metaphysical system, he maintains that not everything that exists is an individual, contrary to what Russell and others have argued. And he gives us examples of things that exist but are not individuals, pluralities and quantities of matter. And as a possible biological example of a plurality, E.J. Lowe suggests that maybe a slime mold we want to think of as a plural entity that's therefore not an individual. And the reason is that pluralities and quantities of matter, he claims, lack the attribute of unity that makes something into an individual. For very different reasons, a similar view has been endorsed by philosophers of physics, including Stephen French, who argues that the subatomic particles of quantum mechanics clearly exist, but they're not individuals, reasons that we need not go into. So notice then that authors who use the term individual this way need to have another, more general purpose term to refer to individuals and non-individuals alike. And typically the term they use is entity. So on this view, an entity is anything that exists, and a subset of those entities are individuals that are characterized by having unity, whereas non-individuals lack that attribute of unity. Okay, then the third sense, the sortally restricted sense, often individual is used in philosophy with an implied restriction to a given sort. As for example, when individual is used to mean individual person, or individual is used to mean individual material object. So if you think in debates in philosophy of social science, for example, people very often debate the doctrine of methodological individualism. An individual in that literature means an individual agent or an individual human or person. So notice that in this sense, individual doesn't denote an ontological category at all. Rather, it just means one entity of a given sort, where the sort in question is typically implicit and can be inferred from context. Okay, what then of the expression biological individual? Well, I take it that presumably this must mean individual, that is biological. That may sound trivial, and indeed it is, but it goes to highlight this to make the point that biological individual is a semantically compound expression, I take it, and not something that has simply been invested with a new meaning entirely unrelated to the meaning of its constituent words. Or more explicitly, from X is a biological individual, I take it that we can validly infer X is biological and X is an individual. That's what I mean by semantically compound. And of course, I mean, this observation is true, but doesn't take us that far in that it's compatible with very many different accounts of what individual means. So let's then ask the question, which of those three philosophical senses of individual, if any, is the operative notion at work in the philosophy of biology literature on biological individuality? Well, what about the logical subject sense? I take it that the answer is no, because you see, this would lead a very large cast of individuals indeed. I mean, things like ecosystems, leaves, telomeres, plasmids would all count as individuals. You can count them, you can refer to them with singular terms. They exist. So clearly that can't be what's at work when philosophers of biology debate what is and isn't a biological individual. What about the unitary entity sense of individual? This may seem more promising and indeed fits with what Sober and Hull have both suggested in some of their works. And Sober argues that an individual is any entity that exhibits integration and interdependence of parts. So again, it seems to be that he's endorsing a metaphysical position a bit like that of E.J. Lowe, according to which an entity is the general category, anything that exists, and some entities are individuals when they're characterized by sufficient unity or integration. And Sober tries to spell out what this attribute of unity or integration that converts, makes an entity into an individual is. In a similar vein, Hull tells us that individual is entity with cohesion. So it looks as if maybe that's what's going on. that people are using individual in this sense of unitary entity. But in fact, I think that is not right. It's really the sortally restricted sense of individual that's at work in most biological and philosophy of biology discussions. And the sorting question, usually but not always, is organism. I'll develop that point in a moment, but firstly I'll say a little bit about the notion of a sortal. So a sortal term, or a predicate, is one that enables us to count the things to which it applies. So simple examples are things like table, pen, chair. Can sensibly say, you know, there are 2 tables in the room, 3 pens, 4 chairs. The contrast here is with adjectival terms, or sometimes called characterizing terms, like red. So Dummett and others have famously argued that in order to grasp a sortal term, to understand its meaning, you need to know, implicitly at least, the criterion of identity for the term, as well as its criterion of application. Whereas for an adjectival term like red, in order to understand it, you just need to know, you know, which things are red and which aren't, roughly speaking. You need to know its criterion of application. Whereas for a sortal term, in addition, you need to know its criterion of identity. And the criterion of identity is meant to be a criterion that settles questions of identity and distinctness in relation to a given kind. And this is one, it's necessary to have a criterion of identity, even if you can't state it explicitly, in order to count under a particular sortal. So I'll give you an example. Take the sortal set. Now, to count the subsets of the set A, B, and C, you need to know that AB is the set AB is identical to the set BA. If you didn't know that, then you would be unable to correctly answer the question, how many subsets does the set A, B, C contain? So that's an example with an abstract object, a set, but the idea is that the same must be true of physical objects, even if it's harder to state explicitly what the criterion of identity is. Okay, now sortals then are a subset of kind terms or predicates, and many biological terms are clearly sortals. Take the term heart or cell or mitochondrion. We can count these entities clearly. We can ask how many cells are in an adult human being, or how many mitochondria in a typical nerve cell or something. But other biological terms are not. So take bone marrow, neural tissue, cytoplasm. We can ask how much neural tissue is in the brain, but not how many, clearly. Sortals and count nouns. Now, the distinction between sortal and non-sortal predicates or terms arises because the world appears to contain stuff as well as entities, to put it rather crudely. Now, of course, it's debatable whether, in fact, all stuff is in fact made up of entities in the way that neural tissue is largely made up of neurons. But that's a metaphysical issue we don't need to get into for now. So you might think then, from what I've said, that a sortal term is simply a count noun. But that's not quite right, for a number of reasons. And an interesting one that I want to focus on was given by the philosopher David Wiggins in 1980. Wiggins observed that many terms in English, such as object, thing, and entity, function grammatically as count nouns, but they're not true sortals in that they come with no associated criterion of identity. And you can illustrate what he meant by just considering how you would answer if someone said to you, look out of the window and tell me how many things you see. I mean, you would obviously be at a loss to answer the question. You would have to say, yeah, but things of what sort? What are you asking me to count? And Wiggins coined the expression a dummy sortal to refer to a term that's grammatically a count noun, but is not a true sortal because it doesn't come with any associated criterion of identity. Let's come back to the word individual. I claim then that the term individual, in any of its usages, philosophical, scientific, or vernacular, is not a sortal term. It's either a dummy sortal or a non-sortal. And that the same is true of biological individual. So I'll now argue for that claim, that individual is not a sortal term. I mean, in the sense of logical subject, this is clear. I mean, there's no way to count logical subjects or things that exist. Therefore, clearly the individual in that sense is a dummy sortal. In the unitary entity sense, it's less clear, but I say also true. I mean, if someone said to you, okay, an individual means an entity that exhibits unity or exhibits integration, I mean, does that really help you answer the question, how many individuals can you see by looking out of the window? I say it doesn't. In the sortally restricted sense, individual is a non-sortal term. So when we use individual to mean, say, individual chopstick, chopstick is a sortal term, individual is not, is merely an adjective. So if individual is short for individual person, then the sortal term is person, not individual. And it follows then that biological individual is not a sortal term either. This follows immediately if we grant, firstly, that individual is not a sortal, as I've just argued, and secondly, that biological individual is semantically compact. Because you see the term biological is clearly an adjectival term, not a sortal term. However, in the individuality debate, it seems to be presupposed that biological individual is a sortal, in that the debate is precisely about how we should count things. So, for example, people in this debate argue about whether an aspen grove is a single large biological individual or should be thought of as a collection of many biological individuals, many individual trees. Or similarly, people ask, when a strawberry plant sends out runners, should we think of that of the growth of a single individual or the reproduction of a new individual? So is there one or two? And this is particularly clear in Ellen Clark's influential formulation of what the biological individuality debate is about. She argues that the debate is an important one because really the role of the individuality concept in evolutionary biology, at least, is to enable us to apply the Darwinian fitness concept, so crucial to evolutionary theory, to problematic entities such as clonal plants. That's why it really matters, according to Clark, how we resolve the question of how many clonal plants we find in an aspen grove, for example. But can we really count biological individuals? Consider the instruction to count the biological individuals in a given spatiotemporal region. Again, you see that, to me, immediately invites the response, individuals of what sort? What do you want me to count? Am I meant to count elephants, flagella, stamens, kidneys, or what? Until a sort has been specified, perhaps implicitly, the instruction is impossible to carry out. But note well, the terms elephant, flagellum, stamen, kidney are perfectly good sortal terms. We can sensibly count the entities that fall under them. We can say how many elephants in the zoo, how many stamen in the plant, how many kidneys in the person, etc. And so therefore we can sensibly talk about an individual stamen, an individual kidney, an individual flagellum, and so on. But notice that these aren't even candidates for being biological individuals in the sense of the current debate. They're just not the sort of thing people are talking about. They're not the problem cases that motivate this discussion. Is it perhaps that these entities fail the test of unity and cohesiveness? I say surely not. I mean, think of a flagellum. On the contrary, it's a perfectly cohesive entity. It's got numerous interdependent parts, like a membrane, basal body, filament, hook, and so on. So in short, cohesion and integration are exhibited at all biological scales. So what exactly is going on? In reality, I think, it's the third sortally restricted sense of individual that's at work in the literature on biological individuality. So biological individual really means individual of a certain biological sort. And the sort in question, most though not all of the time, is organism. I submit that this is the only possible explanation of the joint facts that the expression biological individual is not a sortal, that philosophers of biology believe that they can count biological individuals, but that the count is meant to exclude many entities that clearly do fall under sortal terms. So what the debate is really about, I say it's nothing to do with individuals, individuality, individuation, and that all these terms are better avoided. As you see, they create a false sense of association with other debates in metaphysics that don't exist. Rather, it's really about the concept of an organism. So the debate can be explained quite simply, I think, as follows. We can say, look, paradigm organisms exhibit many features, but then, as well as those paradigm organisms, we find other entities that are only partly organismic. And this can all be expressed without the I word. Because you see, the term organism, I say, is a sortal term, as used in professional biology and in ordinary vernacular English. Now you might say, well, by what rights do I say that that's not a dummy sortal too? Why do I say it's a true sortal? And the answer is simply that it's often perfectly clear how to count organisms. I mean, it's not to say there aren't any problem cases, there are, but nonetheless, it's pretty clear in many cases how you count organisms. If you look at a fish tank and someone says how many organisms are in there, I say that's a perfectly coherent question. How many organisms do you see does not invite a request for sortal specification. Now, of course, there are borderline cases where those are what motivate this whole debate. But the existence of borderline or unclear cases doesn't go to show that organism is not a genuine sortal. On the contrary, all sortal predicates that argue in English admit borderline cases in any natural language. So, for example, ask yourself, is a 20-page pamphlet a book? Well, our uncertainty on that score may complicate the task of counting the books on my bookshelf. But clearly that doesn't go to show that the term book is not a sortal. It's the uncertainty is quite dissimilar from the uncertainty I would face if someone said to me, you know, count the entities in your house. What are the implications of this? Well, I'll trace the implications for three things. Firstly is the idea that we find defended by Peter Godfrey Smith, that there are two types of biological individual. Secondly, I'll come back to the three puzzles that motivated my inquiry to start with. And thirdly, I'll comment briefly on two further aspects of the debate, but won't really have time to get into that. So, Thomas Perdue and Peter Godfrey Smith argue that we should distinguish two types of biological individual. Organisms versus Darwinian individuals or evolutionary individuals, as some authors will have it. An important distinction and very widely accepted, where the former is basically a physiological unit and the latter is a reproducing unit. And they then make the observation that it's not true that all and only organisms are Darwinian individuals. They overlap, these sets overlap, but don't coincide in extension exactly. Now, I say this distinction is fine and important, but I still think there's a logical oddity here. Because you see, the way they set it up, they oppose a sortal term, organism, to a non-sortal term, biological individual. And symptomatic of this is that when Darwinian individual, when Godfrey Smith tries to explain what he means by this term of art, Darwinian individual, he is forced to rely on other sortal terms, such as gene and chromosome. So he says a gene, that's an example of a Darwinian individual. A chromosome, that's an example of a Darwinian individual, but clearly these are not organisms. But let's ask then the question, well, how exactly are Darwinian individuals to be counted? I say there's no clear way of doing that. I mean, to illustrate, suppose you're in the molecular biology lab and a molecular biologist draws your attention to two DNA sequences that they're working on, two token DNA sequences. And let's ask the question then, are they part of the same Darwinian individual or not? I mean, if we can't answer questions of that sort, then we can't count Darwinian individuals. But you see, without sortal specification, there's no way to answer that question, because it's perfectly possible that the DNA sequences could be belong on the same chromosome, so they could be part of the same chromosome, but on different genes, but not part of the same genes, not part of the same gene. So you see, without sortal specification, there's no way of saying whether or not those two sequences belong to the same Darwinian individual. This is indicative of the fact that we don't have a genuine sortal term here. Okay, I began with a number of puzzles. One was, why is biology special? And the answer, I think, is that it isn't. Well, not in this respect. That there's no proprietary individuality concept in biology, contrary to what the literature seems to imply. Rather, we just find individual entities of various sorts, as in all sciences. And one such sort, and a very interesting sort, is organism. And it's hard to define it, are extremely interesting. Now, note well that the type of problem we face here is quite common. It's the problem of providing a principal definition, in astronomy, about what exactly a planet is. Sorry, I'm just going to have to move somewhere else in my house because my internet connection is unstable. These debates, as I see it, are entirely on a par with the debate about whether a termite mound, for example, is an organism. Logically and metaphysically, we just have a sortal concept, and then we have the question, does a particular entity fall in its extension or not? Hull's views. Despite attempting a general individual in the sortally restricted sense in most of his work, when he isn't trying to define it as a unitary entity. It's just that he has a different sortal in mind. So in his doctrine that species are individuals, what Hull is really trying to tell us is that a species is an individual lineage rather than a natural kind. That's the clearest way to express his point, I think. So he's also using the term individual with an implicit restriction to a certain sortal, but in his case, the sortal is not organism, but lineage. Puzzle 3, what about this equation between biological individual and organism? I say that this unfortunate equation arises naturally because of a quirk of the English language, maybe other languages too. You see, the problem cases for the concept of an organism often, though not always, involve entities of which it's hard to say whether they're organisms as opposed to groups of organisms. So think, for example, of the termite mound or the aspen grove. That's exactly the issue there, where the issue is, is this one organism or is it many? Is it a group? And it's very natural to describe this using the term individual, given the established linguistic practice in English of contrasting individuals and groups. So for example, the question, is a termite mound an individual organism? Consider, for example, the question, is a termite mound an individual organism? The key point to notice is that the adjective individual is actually adding nothing here. The question is simply, is a termite mound an organism? To say, does an entity that's a termite mound fall in the extension of the sortal term organism? That's all that the question is. Individual is really only adding emphasis here because we mean as opposed to a group of organisms. This question is entirely on a par, logically and metaphysically, with the question, is Pluto a planet? That astronomers got excited about around about 10 years ago or so. But notice the way we describe that debate. That debate and our uncertainty about how to answer it, the question whether Pluto's a planet, is about the concept of a planet. I mean, you would be guilty of pretentious obfuscation if you were to say that that was a debate about the concept of astronomical individuality. you know, and clearly Pluto is an individual planet. Planets are astronomical objects. So we might say, oh, it's a debate about astronomical individuality, but you don't want to talk that way. And similarly, I think we don't really want to say that the question is about whether a termite mound is a biological individual. It's just a question of whether it's an organism. A brief comment about two other aspects of the debate. One is this, the idea that individuality comes in degrees, which many people have argued for. I mean, this idea is rather hard to make sense of philosophically until you realize that individual is being used in its sortally restricted sense. Because you see, if you thought it was being used in some other sense, then it's natural to say, look, how can something be, is something surely either one or it's many? I mean, you know, being one or being many isn't something that can come in degrees. But of course, it can be indeterminate whether something is an individual S, simply because S is a vague concept or term, where S is any sortal predicate. So that's the obvious explanation, I think, of this otherwise rather puzzling notion that biological individuality comes in degrees. And finally, the word individuation. Much talk in the literature about individuation. So Ellen Clark talks about individuation criteria in plants, and Thomas talks about whether entities should be individuated on the basis of physiological or evolutionary criteria. Now, I maintain that these authors are not using the term in their correct sense. Here I commit a sort of double faux pas by criticizing on a single slide both my own former PhD student, Ellen Clark, and my own host, Thomas Pradieu. But I'd be interested to discuss this point with them more. I maintain that the term biological individuality has led people astray here, in that it's inviting the use of this metaphysical term of art that really isn't relevant. Okay. Just a conclusion. I mean, most philosophers of biology look to science rather than the methods of traditional analytic philosophy. And indeed, I think this is the right thing to do. And it was essential, really, for philosophy of biology to establish itself as a branch of the philosophy of science. However, I think in some cases, the methods of analytic philosophy are still useful, and the debate over biological individuality, I think, is a case in point. Okay, thank you very much.
Thank you very much, Samir. That was very good.
Okay, just trying to organize my slides here.
Okay. I see several questions already. Tyler.
Okay, thank you so much.
So can you just wait 1 second? I'm just going to try and get my kid to turn down the music.
No worries. Always afraid to be the first one to ask a question because it's not entirely well formulated.
Well, you have a few seconds, it seems.
Sorry.
Okay. So you said at the beginning you were worried about kind of doing conceptual analysis because it's sometimes, because it's sort of a faux pas in philosophy of biology anyways. So
well, it might, you know, it might well lead you nowhere, right?
Yeah. Well, I'm going to do something annoying, which is just critique the fact sort of that you're doing conceptual analysis.
Right.
So it seems like a lot of what you're saying turns on whether a term refers to a sortal or a non-sortal, to a count noun or a non-count noun, to a dummy sortal, maybe to an individual or a substance, or maybe a process or something. But I want to maybe argue that it doesn't. The debate doesn't turn on whether something's a count, for instance, or a non-count noun. And my worry there, I think it's a worry that's enough that I could say critique or criticism, is that often whether something's a count noun or a non-count noun turns on something pragmatic about what you're doing instead of about something in the language or something ontologically given. So the example I learned was when you're talking about the number of oranges on a tree, you count the oranges and it's a count noun. So orange becomes a count noun. But if you're making juice, orange is the substance, or if you're transporting oranges across the border, you might say how much orange you have instead of how many oranges, etc. So it seems like something about the biological individuality debate turns on pragmatic issues about what we do when we count stuff, and not whether or not organism or individual is countable.
Well, I mean, maybe I should try and explain what I'm doing here. So what I'm trying to do is not resolve the debate, but try and give a sort of better, more accurate characterization of what the debate is really about, if you like. I mean, I do think that, you know, it's pretty clear that people who talk about biological individuality think that they can count biological individuals, and very often we can. I mean, I think you're quite right that whether something's a count noun or a mass noun is, you know, not. I mean, one and the same term can be both. So, you know, we often give water as an example, or beer as an example of a mass noun, but you could well find yourself in a bar saying, look, can I get 2 beers and 3 waters, please? Or it's implicitly you're referring to a pint of beer and a bottle of water. So, I mean, that observation is certainly valid, but I don't really see it as undermining what I'm trying to do.
Can I briefly follow?
Mm-hmm.
So the sort of where it meets the road, I think, is when people, the justification usually given for wanting to understand biological individuality is that you need to be able to do something like demography when you're trying to figure out whether a population is evolving or changing, and you need to be able to count stuff. But then other people have responded, oh, well, when I want to understand the change in a population, I want to understand something that really I don't need to count anything. It's more like growth or spread, like with the aspens or with mushrooms. And in that context, it seems like the debate doesn't really turn on whether or not you can count mushrooms, or at least some people, for their purposes, think that the debate doesn't turn on whether or not you can count mushrooms.
It doesn't turn on whether you can count mushrooms. Well, I mean, I'm not sure. Maybe the debate doesn't have a unitary, you know, focus, right? There are different issues, but I do think that the question of whether we're dealing with a case of growth or reproduction that's central to some of these issues, I mean, does seem to me to be basically about whether we've got one individual or two. So therefore to be, if you like, about counting individuals. So I mean, I don't really think we disagree. I mean, I don't think what I'm saying is really something that anyone who's got any particular line on this debate should really disagree with. I think they can easily, what I'm trying to say is, look, here's the way it should all be expressed, if you like. And here are the pitfalls of not expressing it in that way.
We have 5 minutes and 3 questions. So I would ask everyone to be super short, both questions and answers, if possible. Let's start with someone who has not yet asked questions. Pierrick.
Thank you. Thank you for your talk, which I enjoyed. So I have questions which, when I followed your talk, it made me think about Alex Rosenberg, who argued in the 70s that fitness is a primitive term in evolutionary theory. And apparently it was not well received in the philosophy of biology literature. And I would like to know if you would agree that if we follow your analysis, then, and the analysis seems compelling to me, that an individual is a kind of primitive term in evolutionary theory, a Darwinian individual.
Well, I suppose what I'm really saying is this, that individual basically just means one. right? So, you know, is it a primitive term? It's a good question. I mean, there are these attempts to define the term individual, both in general philosophy and in philosophy of biology. Like, for example, those who would say, look, an individual is anything that's characterized by interdependence and unity and integration and so on. I mean, I don't have a quarrel with people who want to do that, to use the term that way, as long as they signal that it's a, you know, a specific usage that they're giving. But I suppose what I want to argue is that most of the time when we use the term individual, it's really an adjective. It means one thing of a certain sort, where the sort is generally implicit, because it's obvious what you mean. Yeah, I mean, it's not a million miles away, maybe, from your suggestion of it being a primitive term. Thanks.
John.
Thank you.
John Dupre.
Yeah, I will try and be quick. So that's right. I enjoyed that, Samir, though I must say I'm not convinced, and I still think biology is special. And I suppose what I fundamentally disagree with is you seem to have this idea of organisms as straightforward, countable, sortal terms with a few fuzzy cases at the borders. Mm-hmm. And I think that's what's just, I think that's where you differ, certainly from me, but I think with a lot of people involved in the debate who think there is a unitary entity sense of organism, but it's always problematic. It's right. Problematic. So when you show me your fish tank, I could count the fish, but I'm not counting their internal, their gut microbiota, and how many organisms are there? Are there biofilms that are one, or are there individual microbes that are, I mean, there are many different ways of counting how many organisms there are in your fish tank. And of course, I mean, for me, this connects with the idea that you're not counting individual things, you're counting processes that are more or less interacting, more or less merged with one another. So it's like trying to count, how many tributaries there are in a river. And, that's can become, if you look at an aerial view of a delta or something, you can see that there are lots of ways of doing that. And so what you're doing when you count is you have to specify one of a number of different ways of doing this that's appropriate. And I guess just to finish with a note, I sort of half agree with you, I think, at the end about individuation, that at least that maybe isn't the primary use, but one use of individuation, which I think is very interesting, is as something that biological entities of various kinds actually do, and they do it often by actually collaborating with other entities to provide some stable whole. And unfortunately, the complexity of the different ways they're doing this all at the same time is one of the things that makes for the uncountability of organisms, or the fact that organisms can be counted in many different ways, and we have to decide. So I think sometimes some of the debate looks as if the question is a termite mound an individual, or are termites the individuals? But I think that that's misleading. I mean, that's the wrong way to look at it. The answer is, why do we sometimes want to count termite mounds, whereas we might other times want to count termites, and we might sometimes count their gut bacteria, and we might sometimes not. And there really isn't a good answer to what are the organisms. Sorry, that wasn't very short. I apologize.
Yeah, no, thanks, John. That's really useful. I actually think we agree, believe it or not. So, I mean, yeah, you see, I think that, I mean, if what you're saying is right, then possibly what it goes to show is that the notion of organism, the pre-theoretic notion of organism, is just kind of beyond the pale, really, and isn't of much use when you actually look at the nitty-gritty. And that may indeed be. No, I think that may well be, yeah. So, I mean, when I argue that organism is a sortal term, I mean, yeah, I mean, and I say, look, it's obvious how you count the things. Just look in there and count, right? Then what I mean, perhaps that's true of the sort of vernacular concept of organism that was bequeathed to biology and that biology operated with for quite a long time. But maybe, you know, really in the light of what we now know, that's no longer the case. So the term isn't fit for purpose. But I do still think the question makes more sense to me than the question, you know, how many biological individuals do you see in that fish tank, if you like, in that it denotes a kind of a sword, even if ultimately the problem cases are more numerous than the paradigm cases, if you like. But thanks. No.
I'm sorry to say that we'll have to postpone the final question, Javier. I'm sorry for maybe email discussion because we're out of time. Thank you very much, Samir. That was great discussion. I'm sure we could have continued this discussion for hours. I had at least 10 questions myself, but anyway, we can keep that for later. It's now time to move to our fourth speaker, Kate. Kate Lynch is a postdoc fellow in the Department of Philosophy at the University of Sydney. She has a double background in biology and philosophy. She's the author of many papers in biology and philosophy of biology. Just to give you an idea of what she's been doing in the recent years, she wrote a paper called Heritability and Causal Reasoning in Biology and Philosophy, 2017, with Pierrick Bourrat, who's also with us today. She wrote Interpreting Heritability Causally in Philosophy of Science. Recently, a very interesting and much discussed paper with Emily Park and Maureen O'Malley called How Causal Are Microbiomes, a comparison with the Helicobacter pylori explanation of ulcers in biology and philosophy. And this paper was a target paper in biology and philosophy, meaning that other people, including people who are here today, commented on that paper. So this is a very interesting discussion that you can find in the journal Biology and Philosophy. So Kate, I don't see your title. I guess it's about causality and the microbiome.
Yes, so it's causal explanation in microbiome research. So thank you for that introduction, and thank you for including me in this workshop. Just get my screen going. Okay, as that introduction might have demonstrated, my background is an interest in causation and causal explanation, and in particular, I've worked on causal explanation in genetic research. And I came to looking at microbiome research, first of all, because of the perceived similarities between what was going on in microbiome research and what was going on, at least initially, in genetic and genomic research. So both of these fields, there's this sort of fanfare. It's the hot new thing at the moment is looking at microbiomes, as explaining, you know, all sorts of human health and disease states. And the same sort of thing was happening when people were sequencing the human genome and trying to understand the genome as this book of life. So I have a quote here by Francis Collins, that it is humbling for me and awe-inspiring to realize that we've caught the first glimpse of our own instruction book, previously known only to God. So people have made maybe not such huge claims about microbiomes, but something a couple of years ago that Reset Al claimed is that interactions with, sorry, I just need to move this so I can see, interactions with the microbiome define the human self. Others have talked about the microbiome as being this master puppeteer, controlling our actions and having implications for things like free will and identity. A lot of microbiome researchers think that it's reasonable, in this case from Martin Glaser, to propose that the composition of the microbiome and its activities are involved in most, if not all, of the biological processes that constitute human health and disease. So I see a lot of similarities between genetic research and microbiome research in that they're both studying these very large, complex things with many interacting parts. And the interest is that variability in these entities seems to explain a lot of the variability in human health and disease, in human physiology, behavior, cognition, affect. But what seems different between the two fields is the way that people tend to talk about genomes and microbiomes when they use causal explanations. So in genetics, it's quite common to hear this gene for rhetoric. So the gene for homosexuality was a popular topic a couple of decades ago. The sports gene, mean genes. Now there are separate kind of problems with that gene for rhetoric, but when people aren't talking about the gene for a particular trait or disease, people talk about traits as being genetically caused. So they don't tend to talk about the whole genome. I wouldn't say that, you know, my IQ is in part determined, it's not determined by my genome, it's my genetics. Or I'm short because of my genome, you say I'm short because of my genes. You don't refer to the whole genome as a whole singular entity. This is true both in the way that genetics is expressed in popular science, and I have some examples on the slide here, as well as in scientific publications. But in microbiome research, it's a little bit different. People do tend to talk about the action of microbes and microbial effects, but there's also a lot of talk about the microbiome. And in particular, in human health research, people are very focused on the human gut microbiome. People often talk about the microbiome and mean that human gut microbiome. And as you can see in popular science, people talk about, okay, well, if you're depressed, your gut microbiome must be to blame, or your microbiome is the key to glowing skin. But even in scientific publications, people tend to talk about a mix of it's human gut microbes that are responsible, there's microbial influences, or it's the microbiome. I've got an example here of the oral microbiome. So in this talk, I'm just going to talk briefly about a little bit about causal explanation, in particular reference to proportionality. And I'm interested in the microbiome research cases. What is the most proportional or most appropriate level of causal explanation? Is it appropriate to talk about whole microbiomes, or is a more proportional or appropriate explanation to talk about microbes, you know, clusters of microbes, or the effects of individuals? So proportionality is a dimension of causal explanation that's been invoked in the philosophy of science. And there are a couple of ways to cash out proportionality, but generally what it tries to do is to find a causal description that's not too broad or too narrow. So basically, if a causal descriptor is too narrow, then it excludes important factors that bring about the effect that you're interested in. And if it's too broad, it's going to include factors that have no relevance, no causal relevance to the production of that effect. And so I'm using proportionality in the way that Woodward does in his 2010 paper about genetic causes. And Woodward incites this example of Sophie the pigeon, where you might want to find a causal explanation for why Sophie the pigeon pecks at a particular target. And Sophie the pigeon has been trained to peck at red targets. So you see Sophie peck, and there's a few explanations you could give. You could say that the scarlet target is the cause of Sophie's pecking. And while that's right in some sense, it excludes cases of Sophie might peck when there's other shades of red. So it's too narrow. It's not the most proportional explanation. A more broad explanation could be that a coloured target caused Sophie to peck, but that's far too broad, because if you included some other colours like blue, which Sophie hasn't been trained to peck, then she wouldn't. And so the most proportional explanation here is that the red target caused Sophie to peck. And this can be represented in a diagram like this, where you have a very broad category like colours. Inside of that is a more narrow category red, and inside of that is a much more narrow causal variable scarlet. And in this case, it's that middle causal variable red that's the most proportionate causal explanation of Sophie's pecking. Okay, so to turn to microbiome research now, I'm going to just run through two examples that have come up as very prominent in the human microbiome literature. The first is microbiome, the influence of the microbiome on obesity. And there's been a huge amount of research done on the microbiome and obesity, and I won't be able to get through all of it, but some of the more notable findings. First of all, associations in humans between different kinds of microbiomes or microbiome compositions and human phenotypes of obesity. So We know that microbiomes tend to be more diverse in lean individuals compared to obese individuals, so there's more phylogenetic diversity. And there also seems to be pretty reliable composition differences between obese individuals and lean individuals, and in particular composition changes between these Firmicutes and Bacteroidetes. So you can see on this slide down the bottom right, there's an image there where lean individuals have this overrepresentation of Firmicutes, of Bacteroidetes compared to Firmicutes relative to obese individuals. And these composition changes are also found to change as individuals lose weight through dieting. So that's the rest of this graph is on the far left. You had individuals, obese individuals, prior to starting a diet. And as they lost weight through dieting, the relative abundance of these two groups shifted. So they're associative studies, and it's very hard to do manipulative studies in human populations. So people are using animal models to get to these causal questions about microbiomes and obesity. And most of these experiments involve mice. They are these fecal microbiota transfer experiments where germ-free mice are given microbiome transplants via gavage usually from fecal matter from donor mice. And again, very reliably, what researchers are finding is that the phenotype of obesity is being transferred between mice. So mice that have become obese through diet are then donors for these germ-free mice, which are unusually lean because of their germ-free status. And then they start to gain weight once they gain this microbiome. Remarkably, this has also been shown from transplants from humans into mice. So this study here, Ridjuara et al., looked at human twins where there was a lean and an obese twin, and transplanted fecal matter from these twins into mice. And you can see down the bottom left here that when the microbiomes from, or the fecal matter from lean individuals were transplanted into mice, there was no real change in body composition. These mice were already quite lean. But when they were transplanted in from the obese twins, their body composition changed quite dramatically. There's also been some studies looking at these transfers, so whole fecal microbiota transfers from obese and lean mice, supplemented with particular bacterial species. And this Goodrich et al. Study found that this was a way of not only transferring the obese phenotype, but transferring a lean phenotype, so decreasing weight in mice that had obese fecal matter microbiome transplants. So you can see that's on the graph down here. You've got, these are all into germ-free mice. The germ-free mice were either transplanted with lean or obese fecal matter, but these fecal transplants were supplemented either with this bacterium, which is the plus, or without, and that moderated the effect in the opposite direction. So this is a case where whole microbiomes have been transferred, but in addition to a single microbe of interest. Some other studies that have really narrowed down then on single microbes or microbial groups is Samuel and Gordon and Samuel et al. Focused in on one particular bacterial species and one archaea. And when they transplanted those into germ-free mice, so these mice had no microbiomes except for these two organisms, they managed to increase adiposity in those mice back to healthy levels. And then a more recent study managed to induce leanness and then reverse that, inducing weight gain by manipulating two different types of microbes, Clostridium and Vibrio. Okay, so I mean, there's a lot of information there with those findings. And it looks like that paints a pretty good picture of the microbiome having some sort of causal importance for the trait of obesity. But I want to know is what is the best way to characterize or the best way to causally explain some of these research results? So a very common way that these results are characterized, especially in reviews and in the popular science articles that accompany them, is that it's the microbiome that causes obesity. And you might get that inference from looking at these associational studies where it's these compositional changes in the microbiome are associated with obesity. Though I must say that the associative studies are very careful to talk about associations and not causation in their publications. But there has been some criticism of inferring things about the whole microbiome from those kinds of studies, from the FMT studies, which I'll get to, is that, so the fecal microbiota transplant studies are often invoked as being truly causal because whole microbiomes are being transplanted. But one of the criticisms is that fecal microbiomes are not the same as gut microbiomes. So they're not completely representative of the gut microbiomes that they're trying to manipulate. So this is where in this diagram you might have a slightly different causal variable, which could be a subset, if you like, or slightly apart from the compositional gut microbiome, where you can make a claim about the contents of an FMT, induced obesity. But then some of these other studies are suggesting that what might be really important is just one or a few key microbes. So in those later studies, manipulating just a couple of microbes, with or without an FMT in some cases, was enough to induce changes in leanness or obesity. And so that suggests that perhaps instead of needing to talk about the whole microbiome in this sort of black boxed way, because we don't know what the key microbial components are, there could be some sort of compositional core. Some microbes are much more important for leanness or obesity, some aren't. Obviously, we're not quite at the stage where we know what they all are and how they all interact. But this is akin, I suppose, to talking about, there are some genes for intelligence. We know that there's a high heritability of intelligence. We've identified some genes through genome-wide association studies, but we're not going to say the genome is what makes people intelligent. So I advocate for this more compositional core way of thinking about microbial effects on something like obesity. And in my mind, it makes more sense to talk about the effects of microbes or microbial interactions. And then, of course, there is the issue of the phenotype studied, and this is an issue that you get with any animal model, is that the phenotype of obesity studied in humans is a different descriptor to what's potentially different to what's looked at in mice. So it might be that these compositional associations that map to obesity are different to the effects of FMTs and the core microbes that have been studied in mice. However, given that there's so much overlap in some of these key microbes being identified, these key microbes that have been shown to make a difference in mouse studies being identified as associative in humans, and given that mice show very similar compositional differences to humans in their microbiome differences between lean and obese individuals, I think it's fair to say that they are converging on the same phenotype of the effect. Okay, so what are the researchers actually saying then when they're explaining these results? So these are all passages taken from the papers that I've just used in the previous slides. So Lay et al. Was the study that looked at compositional changes and how they changed with diet. And they use a mix that their title is Human Gut Microbes are Associated with Obesity. But within the paper, they do talk about gut microbial ecology and gut microbial community, so thinking more about the microbiome as a whole. Turnbow et al., it's another associative study, talks about the gut microbiome, again, the microbiome as a whole. Riduara et al., this is the study with transplanting twin human FMTs into mice. Again, just talks about microbiota, doesn't even mention gut microbiota, just microbiota. And then Harley and Karp is a review article about the microbial effects of obesity, particularly looking at fecal microbiota transfers. And they say that the gut microbiome is capable of causally altering development and maintenance of obesity. So they're all the studies that either did whole FMT transfers or looked at associations. So you might think it is a bit more reasonable to talk about whole microbiomes in those cases. But curiously, even in the studies which manipulated single microbes, single types of microbes, there's still this mix of talking about microbes, microbial, and microbiomes. So Goodrick et al. Are pretty specific about the microbes that they manipulated and the way that they describe their research results. Samuel and Gordon in one of their papers are also very specific, but then in their other paper just talk about the effects as being dependent upon the gut microbiota. And then Peterson et al., which looked at this was the study that looked at the Clostridia and desulfovibrio increases and decreases, they talk about the development of obesity as being dependent on the microbiota. So again, the microbiota is becoming this kind of placeholder name for microbial effects, or in some cases, the shown effects of just a few microbes. Okay, I'm just going to quickly talk about one other example, and then I'll wrap up because this is only a short talk. But a second example moves away from the human microbiome stuff and the inferences that come with using animal models. And this is just looking at one particular paper in Drosophila melanogaster. And this is a nice simple study, and this is why I've used this as an illustration, is that what they did is they compared normal flies with intact microbiomes to germ-free flies, and found that when flies were germ-free, so the absence of a microbiome, affected their locomotor ability. So they became more hyperactive, they walked for longer durations, they had less pauses and paused for less time, whereas normally a fly would walk around for a bit, pause, walk around for a bit, pause. So it disrupted the way that they were moving around. And because the Drosophila microbiomes are fairly simple and well characterized, they supplied these flies one at a time with various kinds of bacteria, so various individual types of microbes. And they found that just one particular type of microbe, this Lactobacillus brevis, restored walking to normal levels, to the same kinds of behaviors and levels as conventional flies. So I would think that this kind of study, this should have a very straightforward conclusion, that it's a microbe that's making the difference. At least that's what's been demonstrated here in this paper. And if you look at the proportionality criteria, I think it would make a lot more sense to talk about this single microbe rather than a whole microbiome, or even in this case a causal core, because they did try to eliminate lots of other kinds of microbes in this study as well, which did not find an effect. But instead, it's another perplexing mix of, so the title is a gut microbial factor, which is a little bit of a roundabout way of talking about microbes. Within the text, they talk about select bacterial species, gut bacteria, microbes, and then go on to then talk about the microbiota and the gut microbiome. And this is in stark contrast to a really nice News and Views paper, also published in Nature, based on this research article, which was very specific about the conclusions, where they stated that walking activity in Drosophila melanogaster can be affected by a specific gut bacterium. So they had quite a specific and proportional causal explanation in this case. Okay, so that's basically my beef with a lot of microbiome research, is that I think the microbiome is too often and too prematurely invoked to explain a whole lot of different health and disease, behavioral and cognitive traits. And I think the proportionality criteria can help researchers perhaps make better causal explanations. But one criticism when talking about this with other researchers, and this is where the BMP target article Thomas mentioned, this is where the commentaries have been really useful, is that, okay, maybe there are maximally proportional causal explanations that can be made. There's probably a best causal explanation that you could make for any kind of finding, but all explanation has some sort of pragmatic element, and I've had microbiome researchers tell me this as well. Yes, well, okay, we say microbiome, but we don't mean the whole thing. Of course, it's not every kind of microbe and bacteria involved. We're just black boxing this term microbiome because we don't know exactly which microbial components are important. And so I had one microbiome researcher say to me, it's exactly like how, if you drink a cup of coffee, you might say, oh, this cup of coffee's made me really alert. Well, you know exactly what I mean by that. Really, it's the caffeine that's caused me to wake up and be alert. But there's a pragmatic aspect to saying, okay, well, I drank the cup of coffee and it's the cup of coffee that's woken me up this morning. And I have some sympathy for this, and of course, no causal explanations are going to be absolutely perfect in every way. But I do think that even in the case of the coffee drinking example, for instance, so if I'm to Google something about coffee causing alertness, and I did this, and what I found is a few popular science headlines which talk about actually talking about coffee not causing alertness. that it's just bringing you back up to normal levels of withdrawal, that's all fine. And that's all fine in a normal kind of conversation I might have with someone at work in the morning when I'm having my cup of coffee. But if I go to the actual scientific paper, and this is why I'm looking at scientific communication about causation rather than the science communication literature, which is probably quite easy to pull apart. If you go to that actual scientific article that these news articles are talking about, there's no mention of coffee. They're talking about caffeine. They are specifying this nice proportional causal explanation. And the only mention in this article of coffee is to explain just how much caffeine participants were given, just as a sort of measure for people to understand. And if you try looking at coffee causing alertness or the effects of coffee in Google Scholar, it will automatically autocorrect that to caffeine. It knows that that's the real causal variable that's important. That's the most proportional causal explanation for all these kinds of results. Okay, so in conclusion, what I'm not trying to do here is deny that the microbiome and its components are causally important. There's very obviously something going on in terms of the gut microbiome, at the very least in our guts, in mice guts, in fly guts. It's very causally important for all sorts of health and disease, behavioral, cognitive traits. But what I am trying to say is that terms matter. So depending on the case and depending on the research, it might be appropriate to talk about the effects of microbiome because the whole microbiome, perhaps that is the black box and that's the best you can do. But other times it might be more appropriate to just talk about microbes or microbial communities, or there is some tendency now for researchers to be talking about what they're calling a core microbiome, so causally important parts of the microbiome. I think the proportionality criteria can help identify which is the most appropriate terminology to use for a scientific finding. And while I do think pragmatic considerations are important, and it's very difficult to have perfectly proportional explanation, I think that scientific claims and particular communication in science is worth wording precisely to avoid conceptual confusion. Okay, and that's me. Thank you very much. Particular thanks to Maureen O'Malley and Emily Park, who have worked with me on this, and our BMP article reflects some of this work. To our whole team, Theory, Methods and Biosciences at the University of Sydney, particularly Pierrick Burat, who's discussed some of this work with me, to Rob Knight, and to all the commentators on the Biology and Philosophy article.
Thank you very much, Kate. Okay. Let me move to gallery screen. Yeah, here we are. I already see a question by Javier.
Thank you, Kate. It was an excellent talk. I really enjoyed it. So I'm sympathetic to the idea of proportionality in explanations, and I'm also sympathetic to your response to the pragmatic considerations. But I have another concern. So I'm very sympathetic to attributing microbiome things, because one of the problems that I see by going just to the composition is that you might be masking interactions. Sometimes what you might be having is that interactions between hundreds of microbiome components might be causing a particular trait or a particular phenotype. If in your study you go to 2 or 3 components, which is what seems to be being discovered, you might be masking those interactions that might be playing a very important role. Right, so what would you have to say about this race? Do you see it? Do you share my concern about that?
Yeah, I absolutely see that concern. I think that's an important point that I didn't get to really discuss, is that, so say in the obesity example, there's the effect variable of obesity is very coarse-grained in these studies. So they're looking at, is the mouse lean or is the mouse obese? And some studies are finding that with a whole microbiota transfer. Some of them are finding it with just a few microbes. But it might be the case that a whole microbiota transfer is inducing much more obesity than just these 2 or 3 microbes, right? So there'd be this, how much of an effect do you want to be capturing by either talking about the whole microbiota or these components? So it's an empirical question, really. What really needs to be done is I'd love to see an analysis of these different kinds of cases in the obesity case of the degree of difference in the phenotype measured. And perhaps looking at more fine-grained ways of measuring the phenotypes would help as well. So, I'm not a microbiome researcher. I can't really give methodological recommendations whether people should be starting large and then narrowing down. I think functional approaches, so looking at known microbial effects and microbial interactive effects, are probably a good start. So I definitely see your concern as a concern for a methodological approach to getting those answers in science. So that's why I guess my focus here is more on what the best causal explanation of the current science is.
If I can do a very quick follow-up. Yes, precisely the point. It's also related to when you talk about the gene, about the spores or whatever. In many cases, you can have two individuals with the same gene with completely different capacities. So that my concern comes from there. And I think in the microbiome, for my interest in the holobiome particularly, is much more important, that interaction. So we can keep discussing this in the future. Thank you.
Okay.
Thank you both. We have a question by John. It might be the last one because we have 2 minutes left.
Okay. My question is very similar to Javier's. So if there's anybody else who wants to, but I guess since I'm not hearing that there is. Okay, so yeah, I guess this just started with where Javier ended. I mean, I think first of all, I really thank you. That's a very interesting paper, and I think basically I agree with everything. But there's a point where I was a little unhappy right at the beginning with the way the genome was introduced as a comparison, just in the sense that there might be, I mean, it seems to me there are lots of reasons why you do want to say the genome is the best causal explanation, and you precisely want to resist all the attempt to find the genes for, even if there's a bunch of genes for this or that. And one reason, in addition to the epistasis, is the suspicion of a lot of redundancy, that phenotypes often actually are kind of a tractor state that can be reached through lots of different combinations of genes. And if that's the case, then it's precisely a reason to say the genome as a somewhat integrated whole is the thing that will guarantee the phenotype. Now, to the extent to which that sort of thing is true in microbiomes, I don't know. It was obviously just an empirical question, and certainly I wouldn't quarrel for a moment with the suggestion that one should try and get the causal specificity right. But I think it's more plausible than you sometimes suggested that the causal specificity might be something more like the whole microbiome or the whole genome.
Okay. Yes, thank you. That's very helpful, actually. So I think where we might disagree here is that I would probably agree with you that it might be close to true to talk about the whole genome as being important for a phenotype. But what I'm interested in is explaining variation in phenotype. So I think it's very unlikely that you couldn't say someone's whole genome explains why they are, they have a different IQ to this person, or they're different in height to this person. And so when you're looking at variation in phenotype, I'm looking at the genes that make a difference. And that's where I think talking about genetics, which can still encompass many, many genes and their interactions, is more appropriate than talking about the genome as being the difference maker. Genetics is sufficiently vague that it can capture many genes that you don't know about, and many interactions that you don't know about. And I think in microbiome research, you could do a similar thing. So instead of saying the microbiome, which sort of implicitly assumes that every microbe in there is involved, you could talk about microbes, which could include many, many microbes and their interactions.
Thank you very much. I realized this was very short, so sorry about this. Thank you very much, Kate, for great talk, and thank you for the questions. Again, we can continue the discussion by email.
Thank you.
And we move to the final speaker of the day, the morning for me, and not necessarily for you everywhere in the world. Gregor is our final speaker. Gregor is postdoc fellow in our group here in Bordeaux. Gregor was trained in biology and philosophy. He has a master in molecular biology and a PhD in philosophy of science. And Gregor is the author of several papers already, including one reply to the paper that Kate alluded to or even presented a few minutes ago. And also Gregor is the author of a paper entitled Microbiome Structure and Function, a New Framework for Interpreting Data, which was just published in Bioessays. So the floor is yours, Gregor.
Thanks a lot, and thanks for bearing with us, especially those in different time zones. I hope I can get your attention by talking on one of my obsessions, that is structure and function, and trying to do along the lines of what Samir recommended not to do in every case, but in this instance, I also think it's a useful tool to go into a conceptual analysis of these terms, but with the difference that it's not trying to contribute to merely a philosophical debate, but rather for advancing and addressing scientific problems, in this case, problems from immunology. So I'm not the only one obsessed with the term structure and function. As you can see from this plot here of publications that contain this phrase from last year, but this curve has not flattened in the meantime. If you go into your textbook section, you will quickly see a number of textbooks featuring prominently the terms structure and function again on their covers. So they seem to be very important concepts, but also somewhat in need of clarification, as I will argue. Coming back to a presentation that Thomas and Maël gave on, together with the group in Sydney a couple of weeks back, where they introduced their philosophy in science approach. And as Thomas said about my background, I am an inhabitant of those two worlds. And the aim today will be to give this conceptual analysis to help addressing some conceptual problems in the science of immunology itself. In particular, once structure and function are being mentioned, you quickly run across this slogan that structure determines function. And that has been puzzling me from my days as a first-year molecular biology student and philosophy student. For today, I give you my very own version of trying to give such a philosophy in science approach by first asking the conceptual analysis questions: what do the terms structure and function mean respectively, and what kind of relationships can meaningfully be found between those different concepts. And then we will apply those philosophical results to address some scientific problems in immunology that have to do with immunological recognition and with vaccine development in particular. Also very recent topic in everyone's mind given the current coronavirus crisis. So all of this is either a work in progress or under review right now, so your helpful feedback will be very much appreciated. And these questions will also be the structure of the talk, so let's jump right into it. So structure and function are kind of polysemic terms. Anything that has some sort of order or structure to it can be referred to as a structure, and I will suggest to keep separate at least these four different meanings of structure, either referring to the sequence of a polymer, it could be amino acids or nucleotides. Then probably the most widely used notion will be structure as a three-dimensional shape of a molecule or many molecules attached together on higher levels of organization. You still have the three-dimensional shape of some entities that is important. On a more abstract note, however, organizational structure represented mathematically in the form of networks are often very important to understand the mechanisms behind those structures and their effects. And in microbiome research, as also nicely presented by Kate in the previous talk, here it's often about the taxonomic composition or community structure of microbiomes, represented in pie charts like these or in tables of different percentages of the relative percentage of the phyli being present in those microbiomes. Function, however, that's a philosophical battleground in a way. So we won't have time to go into all the intricate details of the discussion here, but the two classical accounts in the literature still to today are the selected effect versus the causal role accounts of function. Well, basically trying to address different questions like how does something do what it does, and versus why does it do this? So kind of physiological versus evolutionary explanations, but there is a lot of debate going here and we don't have time. For the purpose of linking function to these different notions of structure, I found Arno Worte's distinction of 4 notions of function particularly useful. I'll only use 3 though, that is function as biochemical activity, function as biological role, and I'll throw all the evolutionary selected effect and biological advantages, where Wortes also makes a distinction, into one pot and refer to them as evolutionary selected effect functions, being well aware that there could be much more distinctions made. But I think for putting those 2 pieces now together, but between the notions of structure and function, it's with these, if you buy my distinction here, I think we can make the meaningful connections between the sequence structure of and the 3-dimensional structure of molecules. It could be nucleotides, it could be amino acids in the form of protein folding. But once you have a 3-dimensional shaped entity, this can be linked to its biochemical activity, whereas the organizational structure will then be able to link to the biological role it plays on a higher level in a higher level process or organism. And of course, you can build different levels of organization and take this up onto whatever level you like. Whereas the evolutionary selected effect notions of function address something different that has to do with like a differential persistence or evolutionary changes in the other notions of structure, so I will argue. Emphasizing that we should keep these notions separate and not talk about just functions under one umbrella term either. If you don't like the table overview, here's a bit more graphical representation of the same thing, where we start out with the sequences linking to the 3-dimensional structures, which in turn link to the biochemical activities, and those organized in the organizational structure will give rise to functional roles, and these in turn organized on higher levels of organization have higher biological roles, and so on. So to rationally reconstruct as a result of this philosophical analysis, the slogan that structure would determine function, in immunology, I think it's also important to keep separate these notions of function as biochemical activity in the form of antibody binding, which depends on its 3-dimensional structure, versus triggering an immune response is some biological role that has a lot to do with the organizational structure and is not merely dependent on the 3-dimensional shape of a single molecule, but rather of an organizational higher level entity. Here's a quote from Mark von Regenmortel emphasizing exactly that point, that antigenicity can be reduced to the chemical level and interactions between epitope and paratope, so the binding between 3-dimensional shapes. But for immunogenicity, such a reductive move would be out of place because it depends on many complex biological interactions and organization network of the immune system to then be able to respond as a functional biological role to those threats, pathogens, or other entities and processes that the immune system needs to respond to. I hope you're still with me on this quick napkins overview of the accounts of the relationship between structure and function. Much more could be said, but the aim of this talk is not so much the framework itself, but rather its application to immunology, and that's what I want to spend the next few minutes on. For the question of immunological recognition, we already stressed the importance to keep these different notions separate, that the binding is not the most important part if you want to consider what the immune system responds to. And this has also to do with the textbook view of how the immune system recognizes pathogens and other microbes. Usually they would be dependent on a few family of pathogen-associated molecular patterns, or PAMPs, But it's an increasingly common view to broaden that view, that maybe immune recognition is a bit more broader than we previously thought. So also we are now pushing the line that Thomas Bosch beautifully illustrated about rethinking the evolution of immunity and thinking about structure and function. I think we can also rethink the way the immune system operates in terms of its recognition. So far, it has been a kind of an anthropomorphic biased view where the immune system's main role would have been to fight off pathogens, recognizing pathogens as such based on their structure. But my argument would rather be that what they do, their functional contribution, is more important than which particular microbes are there. I think we will skip this for reasons of time. Again, broadening the view that immunological recognition might include more than just the pattern recognition binding story being a kind of asymptote that we can only fill in some more details. It's a much more broader perspective that is currently going on in immunology. And I think the philosophical conceptual update can be a meaningful and useful contribution in that way. So here are just some illustrations of the different kinds of structure and function immunology here. An antibody where, of course, the binding depends to a large degree only on the paratope-epitope interactions on the three-dimensional shapes, but whether or not this antibody will then create immunity against the pathogen, that depends on the network organization and biochemical activities of the different cell types acting together. This will also be important when we come to vaccinations later. I think time-wise, it makes sense to not go into too more details. If you are interested, stay tuned for the papers or reach out. I'll be happy to discuss. Just to move the view away from these pathogen-associated molecular patterns or danger-associated molecular patterns, I think we can be more broad in conceptualizing any kind of function-associated molecular patterns as well, which would be representative of the roles microbial products play, for example, in digesting short-chain fatty acids. And of course, also sequences can be recognized by kind of the precursors of the CRISPR-Cas being a kind of immune system of prokaryotes. Also an interesting view that other notions of structure besides the three-dimensional binding is important as well. And network structures is here where systems immunology becomes important, where it's not the individual details about what this node represents, but rather the network and its behavior that might be recognized by immune agents and also responded to accordingly on the functional output of these networks. And you can find similar network structures all over the place also in immunology. Versus the taxonomic composition, it would be hard to make a case that the immune system will really monitor the relative percentages of those microbes being present at a given time. They will certainly interact in such a way that will facilitate colonization in those ways, but it would really actively recognize the relative composition. That would be a hard case to argue. And that's not what I'm gonna do. So rather, the immune system would recognize not only three-dimensional structures, but also sequences, maybe even network structures, but not taxonomic compositions. In terms of functions, we don't have the time to go in too many details, just the punchline being that I think it's a possibility and a useful one to think that the immune system might be able to recognize not only molecular patterns, but also functional patterns, observing what is going on in a given microbial host interaction, that it's not so much the identity of the microbes or the products, but rather the biochemical activities that they perform, which can be multiply realized by many different biochemical agents, or the biological role effects that will have on a higher scale. So just to give similarities also to broadening the view of Polly Matzinger, to consider not just the self-non-self distinction, but rather the dangerousness or infectiousness. if we broaden that even more to include also useful activities of microbes and their product, I think this will leave us with opening up this space of not only the molecular patterns that are indicative of certain functions, but rather all the other biochemical activities and roles that can be performed by many different agents. And it's not just a crazy philosopher's idea. Researchers also use those terms of recognition of functional features of microbes, mostly for multicellular parasites or things as allergens and toxins. So the conceptual clarification would then also help to broaden the view as well for microbes, and that here also functional recognition might be at work as well, beyond the just structure-based textbook view. So in a nutshell, we'll focus on these two notions of functions as biochemical activity and biological role that could be recognized by the immune system. Just quickly, case studies. Here is one example of Drosophila protein Persephone, which can recognize a proteolytic activity, so a biochemical activity, which can be realized by dozens of different pathogen proteases. So it's really not recognizing any particular pathogen or its three-dimensional structure, but really the biochemical activity of protease that is being recognized by this sensor. And there's a few more examples which might be a bit borderline, like we talked about ion channels before, where the inflammasome doesn't have a real three-dimensional structure target that it recognizes, rather it responds to ion influx. And for a really higher level physical example, there was this recent study in last year about an immune sensor which can recognize physical force, pressure. So that's clearly something that goes beyond the biochemical level, and it's rather a macroscopic physical force in the tissue where immune sensors exist. And we can easily expect the same might exist for observing and monitoring temperature, pH, oxygen, nutrient availability, where the immune system is crucial in maintaining those levels and responding to those. And in order to do so, it will have to recognize them in the first place. And I think we can expect a number of receptors and recognition entities being able to do that. So to wrap this up, I'll argue that the immune system can, beyond the different kinds of structures, also recognize functions in the form of biochemical activities, biological roles, whereas selected effects will be a different story to explain why the immune system has been co-adapted in such a way that it will tolerate certain roles and activities rather than others, or certain microbes that are prone to perform those activities. It's a different story about the why rather than the how, but it's beyond the page of immunological recognition, I would say. So here's just a repetition summary. I think we can also, in terms of time, just skip to the general lesson that I think that the immune system can be argued that it recognizes some sort of function, and that what is going on might be, in those cases, be more important than which microbes are there, helping to explain why one and the same biochemical agent can be pathogenic or immunogenic in one context and not in another, because it would be dependent not on just the identity and characteristics of those biochemical agents and microbes, but rather what they are doing. Broadening the view that as long as they behave in such a way that the immune system is not affected in a negative way, it will be tolerant. But as soon as they start damaging or like performing detrimentous activities, it would respond. At the same time, that's beyond the view of just negative activities. Why not monitor the amount to which they provide vitamin precursors, the amount to which they help in digesting long carbohydrates, and in maintaining a certain other factors, physical factors, contractions, and other microbial metabolites that are positive beyond the just danger and pathogen fighting way of thinking in immunology. So this really broadens this aspect. And very, very quickly, if you're still with me, I would look at a few bullet points about how this kind of thinking would help us understand how to develop vaccines. Because we stressed multiple times now that binding an antibody is a biochemical activity. And most rational vaccination designs focus on finding a suitable binding partner for an antibody that the host organism has. But these trials then often fail in inducing immunity, because the triggering of an immune response or immunity in general is a biological role that goes beyond the binding activity, which can be modeled onto the three-dimensional shapes of the interacting binding partners. So to provide some meaningful vaccination, we can't vaccine molecules or tissues, but only the whole organism. So we need to take into account the organizational structure and the biological role of the activities of those agents involved. So the general lesson would be that it's not all about the antigens, the three-dimensional structures for developing successful vaccines. I mean, they can be successful in many cases. The current attempts to develop a vaccine against COVID focus on different spike proteins, or in the case of HIV, you have the problem that those three-dimensional structures are a shifting target. So once you have a binding antibody against a structure, it will quickly adapt, change, and again evade the immune system. Same with the seasonality of different flu we have in each season. But what is the more successful approach is to target what they are doing. So the biochemical activities and roles, the way they use to enter the host cells, that can be targeted much more successfully in the case of HIV to prevent those the virus from entering the host cell, whereas the three-dimensional target binding is not always the most successful route, which is not surprising. And in order for vaccines to be successful, only the presence of a three-dimensional structure has not proven to be successful. It always needed to be added with certain adjuvants, which Janeway called the immunologist's dirty little secret. And here again, the mechanisms are still not entirely clear, but it seems that they are doing something biochemically or a biological role in a way that they present the three-dimensional shapes in a context of harming the organism in such a way that it will then trigger an immune response. So we already talked about that. Again, there is some very interesting evolutionary, mostly evolutionary considerations, that it's not so much about the identity of the structures or players or the singers of this particular targets for immunological recognition and vaccination or many other biological processes, but here it might be rather than what they are doing. So the performance on the biochemical and higher levels in forms of the song or the game, that's evolutionarily interesting for being more conserved and also as a target for immunological recognition and vaccination design. My time, I think, is up, so I'll leave it at that. We have the final slide anyway. So conceptual analysis of polysemic terms, a rational reconstruction of the slogan that structure would determine function between three-dimensional shapes, biochemical activities, structure and organization, and biological roles, which I think will help some conceptual puzzles in immunology in terms of recognition and vaccination design. That's the take-home message. And for the philosophical audience, I hope it helped showcase that this philosophical analysis is not only interesting conceptually in itself, but also a useful tool for the scientists working in these fields, I hope. Thank you very much.
Thank you very much, Gregor.
Here's a ton of references.
Great. Thank you. Are there questions? So perhaps I can start. Oh, Javier.
Thank you, Gregor. I mean, you know, I'm very sympathetic to your work. I was wondering, when you make the distinction between what's going on and who is there, one of the things that I think would be nice to emphasize more is the role of horizontal gene transfer in what's going on, right? Because if you think about who is there, you usually use very few genetic markers to determine who is there, but actually bacterial genome includes many more elements, and some of those can be acquired, right? So what's going on might change during the lifetime of the host or might change. So is it something that you plan to do in the future?
So thanks, Samir. That's an excellent suggestion to like how in microbial communities, the ability to be able to do something to change what they are doing is not restricted to have like functionally specialized microbes. So what microbe X can do A, microbe Y can do B, and so on. But they can really easily exchange the capability in times of need or when it's required to pass on some via horizontal gene transfer to the other fellow microbes who might not have been able to perform a particular biochemical task, but now that it's apparently an advantage in the environment, they will quickly jump to sharing those and boost this particular biochemical activity. So indeed, that will help stress that you can start with a different composition of microbes, but once a specific gene is needed to perform, or the gene product rather, to perform a particular biochemical task, they can recruit other microbes and giving them the ability to do the same task, biochemical activity, biological role, to fulfill them at the same time without introducing different microbes, but putting others on the same job, basically. Definitely a good idea to do that. Thanks.
Thank you. Tyler.
Hi. Every time I hear stuff like this about Ford's "It's the Song, Not the Singer," or similar things about what matters in the community, I worry that there's kind of a false comparison going on, like with the particular phylogenetic identities of the community members and their functional roles. Mostly because it's hard to think about how you would compare whether functional roles are more important or less important than community members. So there's lots of things that sort of suggest that, like maybe there's more variation in phylogenetic identity than there is in functional profiles. But I wonder if you could say more about why you think that one is more important than the other at the end of the day.
Oh, thanks a lot. That's a very good point, actually. So yeah, that's very dependent on what kind of question you try to address, I think. So I would only restrict the claim that might be more important for immunological recognition, and in particular the puzzle, why can one and the same molecule be recognized as a pathogen or not as a pathogen in many different contexts, despite being the same entity. So in that case, clearly, who is there remains the same. The difference maker has to be somewhere else. For this particular puzzle, I think what is going on is more important than who is there. But as you rightly pointed out, it might be an unfair comparison. For so many other questions, the right answer lies with who is there, and just compare the composition, and this will be the answer to another puzzle. So I think that it's more than fair to say, like, it's pitting them against each other is not the route to go. Really, they address different questions. Sometimes the answer will be finding out who is there, and sometimes it will be about what is going on. But yeah, the general claim one is more important than the other periods, that's definitely not a good route to go. Thanks.
Thank you. Other questions? We still have time for one question, I guess. Perhaps, Gregor, you could tell us more about the connections with the previous talk by Kate. Do you see, I mean, you alluded to that, but do you see some ways of using the structure-function distinction that you suggested to maybe ask questions about should we care about the microbiome rather than the specific microbes? Can you say a little bit more about this?
So very shortly, I think also I hope that a similar kind of analysis will be helpful for identifying exactly the causally relevant agents, which might be, again, depending on the question you are asking, might lie on some structural or functional aspects, and you might be interested in explaining why a certain taxonomic composition seems to be resolving a health issue, and it might depend on having like functional families in this composition of microbes that are able to perform a task. That could be actually addressed then experimentally if you could have a synthetic community of just biochemical agents, or you have the microbes of one kind with the artificial genes maybe able to perform a particular biochemical task, that this would also result in the same health outcome. But given that these things are so complex, that this is a utopia to think we could have an overview of what kind of genes and biochemical activities are necessary. But maybe for a very isolated case that one microbe is able, for example, in the Vibrio Fisher case, that for producing bioluminescence, if the organism would respond differently if there were no microbes present, but you would still induce the light production by physical means with an electronic wire or whatever, to replace some biological role of biochemical activity with something artificial versus the natural microbial composition that brings about that phenomenon in nature, could help to then tackle, is it really the taxonomic composition? Is it the activities collectively together, or a particular kind of network organization that is necessary to maintain a certain trait or phenotype, also on the microbial level?
Thank you. Any other question, remark, very short? I know everyone is probably tired, not even talking about those for whom it's probably now 6 a.m. So again, I'm very, very impressed by what people can do for individuality, holobionts, and microbiome, or microbes, more specific microbes. I want to really thank everyone. That was great. I really liked it. Thank you so much. And I also want to say again that the next and third meeting will be on June 22nd. The speakers will be Margaret McFarnay, John Dupre, Marie Vass, Adrian Stencil, Vanessa Tribino, and Javier Suarez. And the fourth, and as I said, last one, I promise, will be on June 30. And that one, the final one, would be in the afternoon in Bordeaux. So as I said, more compatible with Californian time, with Rob Knight and Sarkis Mesmanian. Sarkis, incidentally, is the last author of the Schroeter et al. Paper mentioned by Kate. Plenty of things to talk about. So really, I don't want to take more time. Thank you all, and I look forward to seeing you again very soon. Bye.
Thank you, Thomas.
Thank you, Thomas.
Thank you.